Get that up, and hold on, let me finish my organizing. Perfect. All right. So let's go ahead and get started. Any questions before we get rolling today? Besides the obvious one about your exams. Let's turn the chime off because that's going to be annoying now. All right, perfect. All right, so let's go ahead and get started. Today is, I don't know what day it is. Tuesday, April 21st. I think it was the 21st. Tuesday sounds right, yes. And we are on to, thankfully, the last section of this class, which is both a good thing and a bad thing. It is a good thing in that it is the last section. It is a little bit of a challenging section, though. One of the advantages we've had in the skeletal system.
To the class? I know.
One of the advantages we've had to our skeletal system and even our muscle system is if you looked at a bone and you saw that big round head on the femur, it kind of gave you an idea of how it functioned. If you looked at a muscle and we looked at where it attached, what joints it crossed, we could kind of guess at what its function would be. Nervous system isn't going to work that way. It's not as intuitive. When you look at, the part of your brain that moves your arm doesn't look like your arm. The part of your brain that allows you to see does not look like an eyeball. So it is a little less intuitive that way. But I think the one nice thing about it is it'll is a little bit more straightforward when it comes to the anatomy. You have an area or region and it has a function. So 1 and 2, whereas like for instance, the muscles with an individual muscle, there were kind of 4 pieces of information you had to fold in together with each other. It'll be a little bit more straightforward on the anatomy on this that way. And one of the nice things is this is normally an exam we do as a PowerPoint presentation. So again, it isn't going to be like the bones, like the muscles, where normally we should be able to hold those things in our hands and weren't able to do that. So hopefully that will help with that as well. So today we'll start our discussion of the nervous system, talking about the basic anatomy and the things that are going to go along with that. And then we'll also do some histology and talk about those things. There are a lot of assignments to keep you busy. Hopefully you got your pre-labs turned in. If you did not, please make sure to go back and do them and turn them in. It's better to get partial credit for late assignments than no credit for no assignments. And there are 5 additional assignments that are going to be due, starting with the first one, your Unit 12 review, which is due on Thursday. Then you have 2 more unit reviews, both 13 and 14. You have your Physio X. I know we've had so much fun doing those in the last 2 sections that it is going to be so much fun to do it again. And the best part about this Physio X is there are 9 activities. So again, it's due on the 30th. You have a bit of time, but it is not the kind of thing you want to wait till the night before to be able to get it done, because there is a lot. It's not hard, it's just a lot. However, it is one of those things that I encourage you not to start right away. It's what I give it a little bit of a later due date. So we can talk more about the physiology in class, and I think you'll get more out of the Physio X exercises if we do it together. Now, again, if you read the introductions and you read all the information and everything through it, there's certainly enough information for you to go through it, and that might help you to understand the lecture more. But I strongly encourage you, if you're going to get started on it, to read the introduction, to read all the supplemental information that is on it, and not just dive into the activity, to make sure that it makes sense. And if you find you're getting lost, then it's okay to set it down for a bit, and then we'll talk about it as well. The other assignment you're going to have, and it is going to be an assignment that will be graded for correctness, is a 15-point nervous system review. This is similar to our bone review, where we focused on the minutiae of the detail of the bones, and then we put it together into the skeleton. One of the concepts we are going to talk a lot about in this is to basically add to what we've been already talking about. One of the things that we've been talking about in this class, and let me change the color of this to make it and highlight it. Structure, right, relates to function. The function of something affects the structure of something. The structure of something affects the function of something. That is a concept we've been talking about literally since the very first lecture when we talked about anatomy and physiology. However, here in the nervous system, there are really 3 things that are going to go together. And those three things that go together are structure, function, and location. Again, if you think of your bones, all right, while your femur and your humerus are in different locations, a little chunk of the bone in your femur and a little chunk of bone in your humerus do the same thing. Obviously, your pectoralis major and your vastus medialis are different muscles in different parts of the body that move different joints. But if you cut out a little piece of the muscle tissue and replaced them, took a little piece out of the rectus femoris and put it into the pectoralis major and vice versa, they would work the same because muscle tissue does pretty much the same thing everywhere it is. That is not true for nervous system, right? When you point to one point in your brain, that may be the part of your brain that allows you to see vision. Another part moves your hand. Another part allows you to produce speech. So in our nervous tissue, what the neurons do is related to their structure. There are different types of neurons that do different types of functions, but there are also neurons in different locations that have different functions. So the three things we're going to have to get familiar with associating with each other is not just structure and function, but also location. And that's really what that nervous review is about. It's about talking about both the structure, function, and location of neurons. And the nice thing about that is that if you know where a neuron is located, you're going to know its structure and its function. If you know the function of a neuron, you are going to know its structure, and you should have an idea of where its location is. And the same way, if you know the structure of a neuron, you should know its function, and you should have an idea of where its location is. So these three pieces of information are going to go together hand in hand in hand, and you'll see that on the review. I believe I purposely have not made the review available yet because, again, I don't want you working on it yet. It's not going to make sense to you yet. It's also not due till May, so you have plenty of time to work on it. This one, it's like 15 questions each. Some of them can be answered with a single word, right, or 2 or 3 words. Right, there's only a couple that need like a sentence or 2. So it's a little even simpler than the bone review that we did. But again, it's going to really synthesize the information. So you need some of this information as a starting point before we get to that. All right, that is the game plan looking forward. We're not quite done looking back. I have most of your grades, exams graded. I have all of the lecture exams graded, and I am probably 2/3 of the way through the lab exams. So I am working on those. I hope to have those done possibly tomorrow. Based on what I've seen so far, there will be some type of curve. I know that was one of the questions that was asked ahead of time. I have to see what the final numbers look like and curve it. And then also, because this is our format looking forward, I may go back and look at our Lecture 3 exams and see if those need to be adjusted as well. So what I'm hoping to be able to do, and let's go ahead and write this down so that I remember it, and more importantly, you remember it, and it's on the lecture. My goal is to have the exams, have grades, let me say it that way, have grades, have all grades. Updated by lecture on the 23rd. All right, so I'm hoping that by the time we get to lecture on the 23rd, I will have those things available. Whenever the grades are posted, and again, I hope that, and let's put this in parentheses, or after class on the 23rd. If I'm able to accomplish what I want to do, then my goal will be on the 23rd after class, your tests will be available. All right, so when the grades are posted or after lecture on the 23rd, I'm not going to do it before the lecture on the 23rd because everybody's going to be looking at their exams and not listening to the lecture. If I don't have our grades done by Thursday, then whenever it is, my guess would be it'd probably be Thursday night. I'll get it finished, or Friday morning, and then I will get the grades posted then. All right, so either when they get posted or after class on the 23rd, basically whichever comes later, your tests will be available for review. And they will be available for review only 24 hours. I want you to have the opportunity to be able to review the exams, ask questions about the exams, email me. We can have Zoom meetings. We can go through your exam together or things along those lines. But the tests themselves, the questions themselves will only be available for 24 hours for that period of time, at which point they will then be locked and will not be reviewable again. All right, you'll still get the grades. The grades will still be there. And like I said, there will definitely be some, and we'll call it a curve or adjustment to the Unit 4 reviews, from Unit 4 exams. But like I said, I may go back and evaluate the Unit 3 exams for adjustment as well. I don't know yet. Again, I have to look at the final numbers and see how it all looks, decide to whether or not that is warranted. Again, I'm not going to bring them back up to the level of like our lecture exam being worth 100 points the same way that they are. It wasn't an equivalent test, so it's not going to be given the equivalent weight when it comes to that. But again, because of the speed in which we had to get that done, I want to make sure that you are not penalized by that exam either. So I don't want that exam making or breaking somebody's. And again, one of the reasons we leave the points smaller is that should have less impact that way. But I also want to go back once we have these tests in to make sure that it isn't unfairly impacting people's grades. So I will be checking that as well. All righty. So that is the game plan going forward. Any questions on any of that? Dead silence. Excellent. That means everybody's already fallen asleep, or you all completely understand and are excited for the nervous system. Hopefully it's the latter. All righty, let's go ahead and hold on, I'll answer that question. I want to take a picture of the screen so that we have that as available. No, to answer the question, exam 5 cannot be a group exam. No, you will be working in the, so here's what I will say. The format for the lecture exam 5 will be exactly the same as the previous one was, where you will have multiple choice questions, fill in the blank questions, essay questions, and they will all be available at the same time. As I mentioned last week, I didn't just have your exams, I had 431s, my second semester's exams as well. And between both classes, really, the main problems that came up with the lab exam. And again, the one question at a time was a format that is similar to what we do in the classroom. And so I felt it was important that way. I was also concerned about having a test like your test, for instance, where you have 80 images all trying to load at the same time, and you're trying to scroll up and down the screen to be able to see them all. However, the populating of the questions one at a time seemed to cause some problems as well. So I will tell you right now, as quite frankly, as an experiment, I will be doing my 431's next exam, which is before your exam. Their lab exam is a smaller unit with smaller questions. So I'm debating whether or not to make it available like the lab exam, where you have all the questions at once and see if see if that format actually has fewer problems than the one at a time. So depending on how that works in my 431, and again, I'll be honest, I haven't 100% decided that that's the way I'm going to do it, but it is something that I am discussing with other instructors. There is a tremendous amount of discussion going on behind the scenes with other instructors as we're trying to find the best ways, the most compatible ways to give these exams and to deal with this materials. So based on those discussions and my personal thoughts, I may modify their lab exam format. And so then based on what happens with theirs, I will, that could affect how yours is going to be presented. But I do know the lecture exam will be the same. Right, and again, Ashley, you were not the only person who had problems with that. And like I said, with all the exams, most of the problems that occurred, and thankfully there weren't a lot, but the few problems that did occur seem to have occurred with the lab exam. So it is something I am considering doing. But again, my concern is that will cause even more problems or similar problems. So I don't know, we'll have to see, but that is something that's on the table. But other than that, the format for exam 5 will be exactly the same. The other thing you have to remember also is that we also have a final exam as well. So remember, you actually have 3 exams remaining. You have lab exam 5, lecture exam 5, and you do have a cumulative final exam as well. So there is going to be a 100-point cumulative final exam. So you do still have 3 more exams as well as the 5 homework assignments. All right, great questions. Any others? Yes, there is a, no, I don't know the form. The final exam format, it may still be multiple choice. I do not know if that will be, hold on. I got a bunch of questions here. So hold on. I am not certain what the format for the final exam will be, if it's going to still be multiple choice or if we're going to have to change that up. I do not know. There is a study guide that I do present. Again, it is the same study guide that I give for the multiple choice exam, even if it isn't. Really, all it is a list of the major concepts. Again, this is a cumulative final exam. So what I will tell you is that for that final exam, you are responsible for every single piece of information we have covered in this class. So it is not an exclusive study guide where it has everything that you're going to be responsible for on it. You know, is a list of all the materials, because that's basically your lectures and your lecture notes. It is a reminder of the major concepts that will be covered on the exam, but you are responsible for all of it. One of the other things that I want to emphasize, and again, this will be a little easier for you because one of the things I discourage students from doing is using their previous exams to study for the final. Because as you've seen, the final exams, the lecture exams do not by any stretch cover all of the material that was on the exams, right? That it was in that section. And you're not just being tested on what you were tested on, you're being tested on all the material. Now that's more obvious in this one because everybody got different essay questions. So clearly I can't pull just from the essay questions or just from the multiple choice questions to make your exam that way. So you really do need to focus on all of the big concepts. And we can talk more about that final as we get closer to that as well. And so, yeah, I don't know the format of the final exam. Like I said, I've been spending the past week grading exams. No open notes for the final. This is cumulative material that you need to gain. It's open brain. That's it. No open notes. There are no other resources that you are to be using. And then again, remember with that excused withdrawal, again, that is still an option that you do still have. And my understanding is that you have that excused withdrawal up until the grades are submitted. That is what I've been told, but I will emphasize this again. I have not received confirmation on that. So what I would strongly encourage you to do is to go to the website for American River College. I'm sure there's information in admissions and things along those lines where you can find out more information about those excused withdrawals. I don't know what the date is for those. So I can't answer that question for you. I don't know what the final date for the excused withdrawal is. I don't know if it's possible because again, our final date, I don't know where it falls, when grades are going to be due exactly. So all that's going to be slightly different. That would be a risk because again, not only do you need to take the final, but more importantly, I have to have both your final, your lab, and your lecture exam graded and posted before I can submit the grades. So I don't know. I know everything about a lot of things, but I don't know anything about this. The whole online thing is new for all of us. So again, I know it's new and frustrating for you guys. It is equally new and frustrating for us. Trust me, us instructors, just like you guys, bitch and complain and drink alcohol about it, just like I'm sure you guys are doing as well. In fact, we just had a office meeting last night about the online grading. So yeah, again, this is a new process for all of us and we're working through it as fast as we can. And like I said, I can only see so far ahead because like I said, for the past week, I have been furiously grading 4 exams, 2 lab exams, 2 lecture exams. And again, it is an interesting process. But yes, eventually we'll get a better sense of what we're doing moving forward. And again, I've tried to be very open and honest with you guys. As soon as I get information, I'm passing that information on and forward to you. Because again, I want you to be successful in this class, and I want you to be knowledgeable about what's expected of you. I mean, if you look at my syllabus, you know, from the very beginning, it's 4 pages filled with information, because I want everybody going into this and understanding exactly what is expected of them. And I know this has been a challenging experience for you guys, and it has been for me as well. But one thing that I'm trying to continue to do and emphasize is that communication with you guys, so that you know what is expected of you, you know what to expect, and what you still need to continue to do to be successful. Oh, interesting. So CRC. Okay. Here's what I would say to this. I have not heard any information. So again, some of these chats messages are coming in privately, so I know not everybody's seeing them. There was a comment that was made that CRC may be thinking of doing a pass or fail option. I have not heard anything about that. What I would say to that is twofold. There are certain classes that you could get away pass or fail, but what you have to remember is that these classes are still typically prerequisites for getting into programs and things along that. So if theoretically, and again, I know most of you are interested in some type of nursing or kinesiology or something about that. If you're trying to get into a nursing school, then you need to have a 3.0 or 3.5, whatever it is, minimum GPA to get into these programs. So passing this with a pass instead of a grade may give you the units, but it's not going to give you the GPA that you need to be able to get into those programs. So my daughter, who's in high school, is running into similar things, right? Their school district has decided they have the option of going pass fail or taking a grade. But if you're just taking a PE class or something like that, you could take that pass fail. If you're taking an AP class, right, then that grade matters a little bit more. And so you're more motivated to actually get the grade instead of it. My guess is that even if they do allow pass fail options, this is not one of those classes where you're likely going to be able to get away with taking that pass or fail, because this is a class you need a grade in. And also, honestly, if you get I don't know, does a D count as a passing grade? I don't know how that would affect your ability to have the prerequisite to go into 431. But again, I haven't heard anything about that in AR, so I don't know if that's even something AR is discussing, but I doubt it would be relevant for a class like ours. All right. C- is a pass. Okay, well, there are no minuses in community colleges, no pluses and minuses. It's just straight grades because of the transfer protocols. So I guess then, yeah, I guess technically if you got the passing grade, that would get you into 431, because you would have to have a C or better to get into 431. But again, I would be very concerned about how that would affect your ability to get into a nursing program if you took a pass instead of an actual grade. So that would be my concern for them. All righty. But again, that's all speculation on my part, and I don't even know if AR is even allowing that as an option. Any other questions? All right, excellent. Then let's get started. Let's go ahead and switch to nervous system. There we go. And I need that. Where is that? And then where is that? Perfect. All righty. So when we think of the nervous system and the function of the nervous system, as we have already talked about many times in this class, our nervous system, along with our endocrine system, are systems that are specialized for communication. Right? That is what our nervous system is specialized for, right? And that communication allows us to communicate with different parts of our body and plays a vital role in maintaining our health and homeostasis, right? If you think back to their first couple of lectures, when we talked about this process of maintaining homeostasis, we talked about there being a disturbance. You need receptors to perceive that disturbance, right? And then you need that command center. And that command center, remember, as we talked about, receives information, processes that information, makes a decision based on that information, and then sends a signal out to an effector so that we can influence the change that is necessary to bring the body back into balance. And so that is what our nervous system and our endocrine system does. And as we already learned and talked about in our in our last section, stop it. I want to sneak that up and out of my way now. As we just finished talking about in the last section, the way that our nervous system communicates to other cells is by producing a big positive signal. And what did we call that big positive signal again? An action potential. There you go. Excellent. It is by producing an action potential. And again, the advantage of that, as we saw, is that it is fast. An action potential is basically an electrical signal, similar to the electricity in your house. If you flip the light switch, the lights go on. If you flip the light switch, the lights go off. We have that rapid signal being sent along a wire from one location to another, so that when you touch something hot, you pull your hand away from it before you're even aware of the fact that you should be cursing, right? We have that fast, rapid response, and that's what those action potentials provide for us, helping us to maintain our homeostasis. But our nervous system does a lot of other stuff for us as well. It provides us with our perception of the outside world. On the rare occasion we're allowed to go outside, right? Our sight, our smells, our hearing, all of those fun things about that. Our behaviors, our movements, our memories, our emotions, all of these things and more. So our nervous system provides a tremendous, enriching environment of information that basically, essentially makes us who we are. All right. And then, of course, the study of the brain is the field of neurology or the field of neuroscience. All right, let's go ahead and clear this. And let's actually, for this, I want to actually start with our, what do I want? Whiteboard again. Let's go back to the whiteboard. I want to do this. Normally, this is the part where I get to write on the board under normal circumstances. So let's go ahead and do that here. So as we just finished talking about, one of the key, so I'm going to, I need space, so I've usually got my list of participants up, so if someone raises their hand or something, I'm going to minimize that. So if you're trying to get my attention, please either say something or type it in the text of the chat, because I will leave the chat up, but I can't see if you raise your hand or hit yes or no or anything like that right now. So please be aware of that. All right, so what I'm going to do, as we've talked about, there are, as we've been discussing, and this will be, minimize the size of that a little bit. When we talk about the nervous system, there are, of course, structures. And there are also our functions, right? And as we've already talked about, and we know, these two things are clearly going to be related to each other. And I have a capital S in my functions, but oh well. I will somehow survive. All right, let's talk about the functions first. When we talk about the functions of the nervous system, there are really three primary functions of the nervous system. The first, no. That's red. This one I want blue. The first of those are a sensory function. Sensory function is basically where we perceive information about something. This is where we receive our information. And we're receiving that information, put that there, both from the outside and the inside world. All right? So we have this information, this sensory information, that we receive from our information, both from the outside and from the inside world. Again, this is your body temperature. This is, the flower pot you see over in the corner. This is the sound of your kids breaking something in the other room. All of that information, all of those things that we are hearing. As we also talked about, our goal is to then send that sensory information, as we just finished talking about. Hide that over there. Hide that over there. We are going to then send that to our command center. Our command center, as we talked about, is where we process information. And not only do we process information, all right, that we receive, so it receives the information, processes that information, we then compare it to what we know, our base sense of knowledge. Our goal is then to make decisions based on that information. Can you move the names on the side? We can't see it. What names on the side? I don't know why this is still showing up. What do you mean by the names on the side? What is it that you cannot see?
On the side of the screen, it's showing all of the participants for the class.
Oh yeah, okay. Yeah, I don't know why that is showing up. It doesn't normally show up, but oh, there you go. That helps. All right. That works. All right, perfect.
Well, okay, like on my screen, it's still showing. Oh.
Is it gone now?
Yeah.
Can you see my chat window?
It was on my screen. I apologize.
Okay. Oh, okay. So it was yours that you did.
Yes, it was mine.
Ah, okay. Excellent. All right. That I'm happy with. Perfect. All right. Everything else can be seen though, right? Okay, excellent. So our goal, again, of this command center is to process the information. And again, remember, command center is what we called it when we were talking about homeostasis. But really, this goal is really kind of integration. We have this integration of information where we are going to process this information, make sense of information. And this is our kind of command center place. So we, again, and let's emphasize this with a box. So we have this command center. I'm going to go ahead and put a box around sensory as well. Excellent. Where we process this information, compare it to what we know, we make decisions based on this information. And then, of course, our goal is to then send information out. And we send that information out to affect some type of change. And to affect that type of change, we then have our third functional classification, and that is our motor. Our motor is basically where we influence changes. Or more specifically, this is where our effectors are located, right? When we talked about the effectors, these are our effectors that cause the change. And if I remember correctly, and of course I'm teaching the class, so I do, There are basically 4 main effectors, and it's been a long, long time, so I won't put all of you on the spot for that. So I will go ahead and save you that. If you remember correctly, the types of things in homeostasis can be affected, can be our muscles. But remember, as we just learned, there are different types of muscles. So of course, there's skeletal muscle, which is one possible effector. But we also have smooth muscle, cardiac muscle, and the fourth, which is our glands. Great question. So the integration is basically the center part. It receives the sensory information, processes that sensory information, and then once a decision is made, and I'm gonna have to cheat a little bit to do this, we'll bring it down here like that, and then once we bring it down there like that, then we can bring our arrow out this way here to go to our motor. So it is indeed one of our 3 primary functions. Our 3 primary functions of the nervous system is a sensory function, a motor function, and an integration function. Those are the 3 functions. And all of these work together, all right? Again, think of it in simple terms, right? You see something, sensory information. There you are on your back porch listening to the lecture, because that's the best way to do this, right? You've got a nice cold lemonade sitting next to you, and off in the distance in the woods, you see a bear with an axe, all right? That sensory information then travels into your command center where you're going to integrate that information. You process that information, right? You compare it to what you know. Bear, bad. Axe, bad. Bear with axe, very, very bad, right? And so, of course, then you need to affect some type of change. You're going to send signals to some motor function where you realize that you need to stand up, use skeletal muscle, and get into the house, right? Your heart probably will beat faster. Other changes will occur in your glands and smooth muscle and things along those lines. So we have this sequential process. So they're all related together. This is where we get the information. This is where we process the information. And this is where we respond to that information. All right. So those are our functions. And now that I've done this, I want to cheat for consistency. So again, we are talking about the functions. Oops, no. I still want blue. Why are you not blue? Blue. There. Oh, that's why, because I changed it. We have our functions. And like I said, we have 3 main primary, let's say primary types of functions: sensory, motor, and integration. Okay. Those make sense? Again, if we cheat, we can sneak back to our lecture. And we can see the same things here. Nope, not what I wanted. There we go. So again, we have that sensory function where we receive the stimulus, that integrative function, which is our command center where we're analyzing, storing that information, processing that information, and then we send it out to our effector. And again, while a bear with an axe is the perfect classical example, your book uses a more benign one where you see a glass of water, process that information, you're thirsty, or like me, you've been talking for 30 straight minutes, so you grab the drink of water and using your effectors, the skeletal muscle, you bring it to your mouth and take a drink. All right? Those are the basic functions of the nervous system. But of course, also, there are going to be basic structures in our nervous system as well. And let's go back to our whiteboard for this. For structures, we will use red. There are several different types of structures that are going to be formed, that are going to form our nervous system. And of course, primarily of that is our nervous tissue. Our nervous tissue is the primary structure that makes up our nervous system. Right? However, that nervous tissue is comprised of primarily two types of cells. The first of those are our friends, the neurons. These are the functional cells. These are the ones that produce the action potential. These are the ones that send the information. These are the ones that do all of the heavy lifting that we need to be able to do. And the second are what are known as glial cells. They're also known as neuroglial cells. And those two terms are completely interchangeable, the same way that muscle cell and muscle fiber were interchangeable. Glial cell and neuroglial cell are completely interchangeable as well. And let's do it this way for consistency's sake. AKA neuroglial cells, and these are our support cells. Their job is to help those neurons to do the jobs that they need to do. All right, so those are the structures. Now, as we've already seen, and I will do a very, very simple basic drawing of this because we've already talked about it. And again, we'll get into much more detail and talking about the anatomy in a bit. But if you remember, we have talked a little bit about our neurons. Oops, you go there. You go there, you go there. Our neuron basically has two main structures, or two main components. It has the body, right? Whoops. For consistency's sake, let's keep it blue. It has the body, and as we've also talked about, it has the axon. One big long axon. And again, we know it ends at axon terminals. It is going to have synaptic end bulbs. They're going to have voltage-gated calcium channels. We've talked about this anatomy a bit as well. The reason this is important is because the structures of the nervous system are going to be comprised of either neuron cell bodies or neuron axons, or in some cases, both. Right? Let's start easy. When we talk about specific structures, the nervous system, what are the structures of the nervous system that come to mind when I say that? What are the obvious structures of the nervous system that everybody should be able to figure out? I'll wait. Think of your nervous system and tell me a structure of it. Okay, nerves are good. Synaptic end bulb is a part of the neuron. Absolutely, we have that. But come on, when I say nervous system, what's the first thing that comes to mind for you? There you go, brain, absolutely. What's the second thing that comes to mind for you? Spinal cord, perfect, excellent. Those are the ones that I want. Brain and spinal cord. Those are our two biggest structures. They're the most obvious structures, right? And again, we'll talk a lot about those. But there was a couple other things that were put on that list as well. One of those is a nerve. Now, as it turns out, there are nerves, and nerves are basically bundles of axons. So you get a large bundle of axons together, and they form what we call a nerve. All right. Now, there are two types of nerves. The first are cranial nerves. Based on a name like cranial nerves, guess what structure these nerves come off of? They come off the brain, absolutely. And there are 12 pairs of them. you are going to learn everything you ever wanted to know about the cranial nerves and more. In fact, as much as 25% of your lab exam, depending on how much of the sensory stuff we get to, as much as 25% of your lab exam could be on your cranial nerves. It is going to be a huge, huge part of this section. The second type of nerves are spinal nerves. Guess where spinal nerves come, what structure they come off of? Spinal cord, there you go, exactly. And as it turns out, there are 31 pairs of those. So we are going to have nerves, which are going to be bundles of axons. We also are going to have bundles of, let me see how I want to do this. Need an extra space there. Of cell bodies. And one of the ways that we get a bundle of cell body is something we call a ganglion. A ganglion, oh, helps if I spell it right. A ganglion is a cluster of cell bodies. But there are other structures as well. One of the things that happens is, if you think about it, our brain and our spinal cord are big collections of neurons and cell bodies and those glial cells and all of those types of things. It's a big, huge amalgamation, a big, huge mess of all of them. But we have stuff like that outside of the brain and spinal cord as well. There are parts of your body that have big, elaborate networks, bundles of nerves, bundles of ganglion, and basically they're like little mini brains. Of course, we don't call them mini brains. The special term we have for them is something like a plexus. A plexus is an elaborate network of nerves and ganglia. Ganglia is the plural, ganglion is the singular. Probably one of the classic examples of this is in your digestive system. Your stomach has an elaborate network called the enteric plexus. And it's commonly, affectionately referred to as your gut brain. Because here's what happens. You sit down and have a big cheeseburger for breakfast. And when you have that big cheeseburger for breakfast, it goes into your stomach and your stomach stretches. When your stomach stretches, there are stretch receptors in your stomach. Those are our sensory structures that perceive that information. They then carry that information to this elaborate plexus, this bundle of nerves and ganglia within your stomach. And your stomach says, hey, there's food inside of me. I need to do something about it. And so it sends a signal to the smooth muscle of your stomach to start churning that cheeseburger. It sends a signal to the glands of the stomach to start releasing hydrochloric acid, right? And that whole process, getting a stimulus, processing the information, making a decision and affecting the change, all happened right there in the stomach. That plexus was able to do all of that on its own. Now, again, it's not making very complicated decisions. Stretching means, make acid and contract is pretty basic decision, but still these plexes allow us to be able to do that. And then, of course, the other component are our sensory structures. Oops, did I spell that right? Right, sensory structures like the light sensing cells in your eyeball, the smell sensing structures in your nose, your taste buds, all of those skin receptors we talked about, the Pacinian corpuscle, the tactile corpuscle, all of those sensory structures are things as well. Now, these is a partial list. We'll add one or two more things to it. This partial list. And these structures of the brain are organized as well. With the structures of the brain, there are two main divisions. And I'm pretty certain you can identify what those two main divisions are. They are the central nervous system and the peripheral nervous system. So when we think about those two main divisions, we have our central nervous system, and it is our central nervous system that is responsible for making the decisions, for integrating information, for being that command center. And then we have the peripheral nervous system that is basically the pathways of information, our sensory information coming in and our motor information coming out. And I don't have a dotted line, so I will cheat and use my highlighter to put a line right down the center there. And that is a horrible line, but that's okay. It serves our purpose. And when we talk about these structures of the nervous system, they are going to be divided between the central and the peripheral. And of course, what structures are the structures of the central nervous system? Exactly, are the brain and the spinal cord. Those are the structures of the central nervous system, and therefore the rest of those structures, the nerves, the ganglion, the plexus, etc., all the rest of those things fall in the peripheral nervous system. And let's go ahead and name them. Nerves, ganglia, plexes. Plexes is the plural. Oops. Plexes or plexuses, I don't know what it is. Sensory structures. Let's say it this way, special sensory structures. And they don't even have to be special, just sensory structures. We'll do with that. Those are the things that are in our peripheral nervous system. And I think if I put that there, that is good. And I can sneak that back here, and that works. All righty, excellent. Now, now that we've made this division, there is one more thing I want to highlight, and I'll do this in pink. Nerves, notice, are only a component of the peripheral nervous system. All right. But our brain is going to have bundles of axons as well. So in our central nervous system, a bundle of axons is called a tract. So we will see tracts in our central nervous system, and they're basically bundle of axons. So those will be in our brain and our spinal cord. And it's the same thing with bundle of cell bodies. This bundle of cell bodies are called ganglia in our peripheral nervous system. Pardon me, yes, peripheral nervous system. Whereas a bundle of cell bodies and switch back to pink. In our central nervous system, a bundle of cell bodies is called a nucleus. Excellent. So again, when we think of our brain and spinal cord is going to have nuclei, the plural. It is going to have tracts. And there is two other important structures that are found in our brain and spinal cord as well, our gray matter and our white matter. Oops. Gray matter is comprised of neuron bodies, and our white matter is made of axons. So these are the structures that form our divisions, our functional divisions of the nervous system. All righty. Nope, don't want to do that. I wanted to grab this. I can, perfect. With me so far? Because it's just about to get worse. So if this isn't making sense now, ask now, because like I said, it's about to get even worse. Or it's not really worse, but it's going to get. We're adding more layers. All right. Then if this is okay and we're comfortable with this, let's move to the next part. The next part involves, what color should we use for this? Let's go ahead and use brown. No, we use orange. It'll stand out better. Notice when we talk about our functional divisions, we said there are three primary types: sensory, motor, and integration. But there are also specific types as well. And with sensory, there are three specific types of sensory information. That we need to be able to talk about. The first of these is what we call our somatic sensory. Somatic sensory is basically general information on the outside world, right? These are the things that we often and mostly associate with the skin. Cheat and sneak that up there. If you think of things like touch, pain, tickle, itch, those types of things that we talked about, those are general sensations, and they can be felt pretty much everywhere all over the body. Right? So again, those are our somatic sensory information. But we also have what is known as visceral information or visceral sensory. This visceral sensory is what is going on the inside of your body. Or let's say this way, info from inside a body. This is often information you're not consciously aware of, right? Can you tell right now, I'm about a quart low of calcium right now, or my pH is a teeny % off inside my body? No, those are things that you cannot be consciously aware of. But is knowing the pH of your body, knowing how much calcium is in your blood, is that important information that we need to monitor and keep track of? Absolutely it is. And so to do that, it is going to use special sensors inside the body that we call Entero Receptors. And these Entero Receptors tell us the information about what is going on inside of our body. And this is our visceral sensory, or what is also sometimes referred to as autonomic. Actually, I don't want to put it there. Let's put it at the end. A, autonomic sensory. Lastly, our third type of sensory information is our special sensory information. This is specific information from typically from a specific special sensory structure. Hearing is a great example, right? You cannot hear with your feet or your elbows or your knees or your tip of your tongue. You have special sensory structures inside your eye, inside your ear, that allows you to perceive this information, all right? I am telling you not to do this, okay? Do not do this. In fact, if I ever catch you doing this, I will take participation points away from you. Do not do this. It's a thought experiment only. Do not do this. But theoretically, what happens if you rub your eyes a little too hard, right? You wake up in the morning, you have all that gook in your eyes, so you give them a nice vigorous rub to wake up, or you're really tired studying for this and you give them that really vigorous rub, which you should not do. It can damage your retina if you do that. Do not do that. But theoretically, if you do that, right, theoretically what would happen is you would see a little bit of white light. Because someone's shining a white light in your eye? No. But what's happening is you are putting pressure on your eyeball. That increases the pressure inside your eye, and that increased pressure squeezes the retina. The retina is the special sensory structure in the back of your eyeball. Now, squeezing it can damage it, which is why you should not do that. But when you squeeze your retina, what ends up happening is it gets stimulated. And it is a special sensory structure. So when it gets stimulated, it fires action potentials. It sends those action potentials to your brain, and your brain thinks it's seeing light. Well, really all it is you've got an increased pressure on your eyeball, right? So things like vision, things like taste, things like smell, things like hearing, right? Those senses, balance, equilibrium, balance, those are our special sensory structures, that information, and they are provided by special sensory structures. So those are the three branches of our sensory, the three specific types of sensory. And if there's three specific branches to our sensory, or three specific subdivisions, guess how many there are to our motor? 3 as well. There are, and for this I'm going to use, I'm going to use the lighter green for this one. No, wait, hold on. Need to change that back to orange. Perfect. New line. That was new line, light green. Excellent. There are indeed also 3 specific subdivisions to our motor as well. Although really, there's 2. There's 2 specific subdivisions, but one of these subdivisions has 2 subdivisions of its own. And we'll see that in just a minute. And again, this is information you probably don't want that. Excellent. And then I'm going to cheat and grab this and move this over here and do that to it. Perfect. So there are kind of 3 subdivisions, but we'll see how this works. The first subdivision is what we call our somatic motor. This somatic motor subdivision should sound familiar, because this is the one that we know controls our skeletal muscle. This gives our voluntary control, and it gives us that voluntary control of our skeletal muscle. So when we were talking about our skeletal muscle and controlling it at our neuromuscular junction, it was a somatic motor neuron that provided that voluntary control. Oops, and somehow I lost my 1. There we go. Excellent. The second subdivision is what we call the autonomic motor. And the autonomic motor division is what gives us our involuntary control. And it gives us that involuntary control of our smooth muscle, our cardiac muscle, and our glands. However, as you likely know, there are 2 subdivisions of this autonomic motor. The first is the sympathetic, and the second is the parasympathetic. we will talk about these, and I'm going to cheat and give myself a little bit more space by putting these down. The sympathetic and parasympathetic nervous system has some cutesy little mnemonics that are often used to be able to remember them. Our sympathetic is often referred to as our fight or flight. And again, that's what we use. Like I said, when you see that bear with an axe, right, you have two choices. You can fight that bear with an axe, or you can outrun somebody in the class, right? You have two choices on how you're going to deal with it. And fight or flight is actually a pretty good mnemonic for remembering all of the functions of the sympathetic nervous system. Parasympathetic is commonly referred to as rest and digest. That mnemonic isn't quite as good. There's a lot more that goes into our parasympathetic than just resting and digesting. But for what we're doing now, it's a good starting point. Our last 2 weeks of the class, we will talk almost exclusively about our autonomic nervous system. So again, our autonomic nervous system is one of those things you're going to learn everything and more you wanted to know about. And I will tell you that it's going to be rough. We've had a rough semester. Normally more rough than a normal semester will be. But I will tell you right now, those last 2 weeks when we're talking about the autonomic nervous system, that's the information that is probably going to be most important and most helpful for you when we get to 431. Because if you think about 431, whether it is the cardiovascular system, where there's cardiac muscle in the heart and smooth muscle in the blood vessels, or the respiratory system and its smooth muscle, or the digestive and its glands and its smooth muscle, every organ system we talk about in 431 is going to be controlled by the sympathetic and parasympathetic nervous systems. So this autonomic nervous system, which again, it's the very last thing we talk about because it's the thing that really leads us into 431. And therefore the most important for it. But it's the last thing, so often people are fatigued and tired by them, but it's probably the most important thing we'll talk about in this class. This and the tissues. Autonomic nervous system and the tissues are the two things you'll use the most in 431. So those are the things you need to make sure you truly master. All right, so there you go. Look how amazingly beautiful that is. We have looked at the structures, we have looked at the functions, we have looked at the functional subdivisions, we have the relationship of them. This is the organization of the nervous system, both the structural and functional organization and how they work together. All right, excellent. I like that. Let's take a pretty picture of this. And with that, let's sneak back to our lecture where it shows us the same thing. So like we said, billions of neurons, trillions of those glial or neuroglial cells from the brain, the spinal cord, our cranial nerves, our spinal nerves, our ganglia, our sensory receptors, our plexes, right? And as we also know, our tracts and our nuclei. And they all work together to form that organization. And as we talked about, we have our central nervous system where we're doing our integration, and it's our command center, and that is the brain and spinal cord. Our peripheral nervous system, which is our lines of communication. And that's one more thing that we need to add to our list. We'll get to that in a second. Our nerves, our ganglia, our sensory receptors, our plexes, things along those lines. That's the part right here. This is what I wanted to add to our previous picture. So I'm going to add to the picture and then take another, add it to our whiteboard and then take another picture of it, is the directionality of these things. So it says it here, but let's go back and do it on our whiteboard. Information, what color haven't I used? We'll use purple. Information coming into our central nervous system. So like an arrow going into our central nervous system. So our sensory information is what we call afferent information, or it's the afferent pathway. Right, going, because it is going into the central nervous system. And I think I can fit that around my arrow. There we go. And the information coming out is efferent info. right? Or the efferent pathway, and that is out of the central nervous system. There you go. So those are the directionalities of it. Sensory information is afferent, going into the central nervous system. Information coming out of the central nervous system is efferent, and it is motor. There you go. Excellent. So now I got to take a new picture. Save the whiteboard. Go back to lecture. Right, and then as we said, there are functional subdivisions of the sensory, our somatic motor, somatic sensory, general sensory information, temperature, pain, tickle, itch, a lot of those things we get from the skin. Our visceral or autonomic sensory, that's what's going on the inside of the body, and those special receptors. And like I said, we have subdivisions of the motor neurons as well, our somatic motor, which controls our skeletal muscle, our autonomic motor, which controls our smooth muscle, our cardiac muscle, and our glands. And as we talked about, there are two subdivisions of the autonomic, and those are the sympathetic, our fight and flight, and our parasympathetic, our rest and digest. A lot of words, both on the slides and on my whiteboard, but your book does have a pretty picture that does an almost perfect job of showing us all of this. Notice, and I need my annotation thing back for this. Here it shows the divisions from a structural standpoint. We have our central nervous system, which is the brain and the spinal cord, and the peripheral nervous system, which are the nerves and all the other structures that are out there. Notice we have our somatic sensory coming in. We have our autonomic sensory coming in, that visceral sensory telling us about stuff in the stomach. We have our somatic motor out. We have our sympathetic, for instance, increasing the heart rate, and our parasympathetic affecting and controlling the bladder. So we show our autonomic branches here. Now it's important to know sympathetic goes to the heart and as well as parasympathetic, and we'll talk more about like that I said. So notice the only thing we're really missing, and thankfully we can now draw it in, is our third afferent pathway, and I guess that should be light blue to match the other one. There we go. And now I can erase that one. And for this, we need something super special, like, I don't know, how about an eyeball? There's our eyeball, and oops, that eyeball, and that eyeball there gives us that special sensory information. Information that is, again, afferent sensory information coming in, and now our picture is perfect. So there you go. That is the organization of the nervous system, both from a structural and a functional, and not surprisingly, how the two work together. All righty. Any questions on any of that? All right, excellent. That was a tremendous amount of information. So let's take some time to process it and make some sense out of that. So we'll go ahead now and take our first break. I know it seems like we went a long time, but remember, the first half an hour I basically spent talking about the introduction to this and talking about our future and our past exams. So I haven't been lecturing too long, but still, we'll go ahead and take our full 10-minute break. So come back. I guess that'll be 1:25. Will be our restart. And at that point, again, I will start the recording again. All right, so any questions before I stop the recording and we take our break? Going once, going twice. All right, see you in 10 minutes. All righty. So We've talked about both the structural and functional organization of the nervous system, talked about the subdivisions, talked about the relationship of all of those. So what we need to do now is talk a little bit more about the anatomy, and in particular, the microscopic anatomy. As we mentioned, nervous tissue is basically comprised of two cell types, or two types of cells. And again, as we said, those are the neurons and the neuroglial cells, which is what it says right there in front of you. So let's talk about each of those. As I mentioned, our neurons are primarily our functional cells. These are the ones that produce the action potentials and spread the signal. These are the ones that communicate and do all the other important things that we talk about. They're able to do that because when we talk about their characteristics, one of the big characteristics that they share with muscle cells is that they're irritable. Of course, as you remember, irritable means to receive a stimulus and respond to it. And our neurons do it in the exact same way. They respond by producing an action potential. So we could also say, like we did for muscle cells, that they are electrically excitable. Right, that is definitely the same. So they're electrically excitable. However, another important characteristic that neurons have, and is also one that it shares with our muscle cells, is conductivity. We didn't emphasize this on our muscle cell, but we did talk about it. Because remember, as we mentioned, that muscle cell produced the muscle action potential at the motor end plate. But then it spread down the sarcolemma and down the transverse tubules. And as we said, that sarcolemma was specialized to produce and project an electrical signal. And the same thing is true for a neuron. By conductivity, it means that it can produce and propagate. That's the big fancy word that basically just means to send from one place to another. Oops, if I spell another right. To produce and propagate an action potential. Think of it this way. If you had one of those old-time wooden yardsticks that had the metal ends on that, you could take that metal end and you could jam it into a light socket and you'd probably get a decent-sized spark. But if you were holding the other end of that wooden ruler, you wouldn't have a problem with that because that wood does not conduct electricity and you wouldn't be shocked by it. However, if it was one of those metal yardsticks, you jab one end of that and you're going to get quite a jolt at the other. So that ability to be able to send a signal from one part of it to another is super important, especially when you think of your nervous system. You have neurons whose cell bodies are located in the small of your back, and they need to send information all the way down to your big toe. They need to get information all the way from the big toe. So if you step on a Lego as you're walking around in your house, that excruciatingly painful signal needs to be sent to your spinal cord so it can go to your brain and tell you that you're supposed to be cursing right now. Right? So that ability, and again, that's about 1 meter in length. So being able to send that signal long distances is one of the important functions of that, and that's really the key. It allows the cells to send the signals long distances. And those are some of the key characteristics of our neurons. Now, the other kind of cells are our support cells, our neuroglial cells. And as I said, we're going to think of them as support cells. We're going to talk about them as support cells, and they definitely do serve a support function. But I will also tell you that there's a lot of really interesting information that is coming out and has come out in the past few years about these neuroglial cells. And so our understanding of them is changing dramatically. Again, if you take this class 10 years from now, and quite frankly, I don't know why you would want to, but if you did, we may be seeing a different story about the neuroglial cells. And one of the classic examples of why this occurred is because of an individual by the name of Albert Einstein. Albert Einstein was a brilliant individual that I'm sure many of you have heard of, or at least own an E equals MC squared t-shirt. Super smart. And eventually, after a long, boring story, neuroscientists were able to get a hold of his brain after he died. And they were all super excited to be able to cut up his brain and look at it under a microscope so they could see what kind of amazing interconnections of his neurons that he had that allowed him to make these massive leaps of cognition and understanding that mere mortals like you and I are not capable of doing. And so they cut up his brain and they looked it up under a microscope, and what they found is the number of neurons he has, the organization of the neurons he has is exactly the same as you and me. So call up grandma, call her on the phone, say, hey, grandma, guess what? I have the exact same neural network as Albert Einstein, and she'll be really impressed, and she'll send you $20 in the mail. However, it turns out his brain wasn't identical to a typical brain. There were some areas of his brain that had as many as 4 times as many neuroglial cells. Others that had 2.5 to 3 times as many neuroglial cells. So this led us into a direction to maybe think that maybe while these neuroglial cells are support cells, maybe they do play more of an important role in storing information or processing information or something along those lines. And it really opened a whole new field of study. So like I said, there's a tremendous amount of time, effort, and money going into it right now. But for right now, we'll focus on the things that we know for certain, and those are the support roles of those neuroglial cells. The other important thing I want to emphasize is there have been over 100 different types of neuroglial cells that have been identified, right? You are not going to be responsible for all of them, not even close. There's like 7 that you are going to be. You're going to be responsible for the 7 most common neuroglial cells, know where they're located, and know what they do. All right, let's talk about these. Starting first with our friend, the neuron. Here we see a picture, almost as good as the one I've been drawing again and again and again and again for us. As we have talked about, this neuron, and I will use black for this, has a large centrally located cell body. And that large centrally located cell body is what we, and of course now the picture has changed on me. Actually, we'll start back here. Large centrally located cell body, and then it has processes coming off of it. Notice there are 2 main types of processes. There are the short, elaborately branched processes, and there is this long, singular, unbranched process. We talked about this when we were talking about the muscle systems. These short, elaborately branched are what we call the dendrites. And that one single unbranched one is what we call the axon. That axon is the specialized part. That axon is the one that can be up to 3 feet in length. And it is single and it is unbranched. But as we also talked about at the end, it does eventually branch into those axon terminals. And as we know, it ends at those specialized structures that we're calling the synaptic end bulbs. Again, there are different terms that are used. All of them are appropriate. I have probably a half dozen different terms that have been used for these. Sometimes the axon terminals are known as the teledendria or things along those lines. These are the synaptic boutons or synaptic terminal ends, or end buttons, all sorts of fun terms. But for consistency, I will use the term axon terminal to distinguish the parts where it branches, and synaptic end bulb for the specialized ends that we learned about in the muscular system. So let's take a closer look and add to that anatomy, which we've already learned. Starting first here, where is my mouse? There it is. With the cell body, and as I mentioned, the cell body is where we have the nucleus, where we have the mitochondria. Here, look, they've got a Golgi apparatus in here, all sorts of fun things, the kind of things we expect to see inside of a cell body, and that cell body is known as the soma. However, these cell bodies are not completely identical. There was a very interesting thing, and I'll use dark blue in my highlighter to emphasize this. When they first started looking at these under the microscope, what they found is this cell had a massive amount of this really dense material inside of the cell body. They didn't know what it was, but they did know that there was a special blue stain that they would use that really made it stand out very elaborately. And because it was the blue stain that made it stand out, and that blue stain happened to be called a Nissl stain, these structures were known as the Nissl bodies. Now, as our microscopes got more sophisticated, our understanding got more sophisticated, what they began to learn is that these things that they were calling the Nissl bodies were a bunch of flattened, interconnected membranous sacs, kind of like the ones you see here. Here's a big flattened interconnected membranous sac with all these dots on the outer surface of it. What does this look an awful lot like? What is this stuff that I'm putting the circle around? It's a cumulative class, so you guys should be able to figure that out. And not just any endoplasmic reticulum, but which one? Rough, perfect, absolutely. It turns out what these Nissl bodies were they were the rough endoplasmic reticulum. And if you remember, rough endoplasmic reticulum is what we use to make proteins. Oops. Remember, this cell releases that chemical signal way down at the synaptic end bulb. Those are all proteins. And this cell has an elaborate cytoskeletal structure. So neurons need to make massive amounts of proteins. So it's not surprising that it has a very dense, large amount of rough endoplasmic reticulum. And so we can call it both. We can still call it rough endoplasmic reticulum, or we can refer to them as the Nissl bodies. Both are acceptable. As I mentioned, it has an elaborate cytoskeletal structure. To have all these processes sticking out of it and to have them going down the axon, we need cytoskeleton. Remember, we talked about actin filaments make them dynamic. Those intermediate filaments give it stability. And our microtubules allow for movement. Well, remember, if those neurotransmitters are going to be transported a meter down the axon, we need lots of myofibrils to be able to do that. So all of the elaborate cytoskeleton, and that's really all it is. Our neurofilaments are just our cytoskeleton. I'll cheat and write it down here. The cytoskeletal structures of a neurons are basically protein fibers, so they're called neurofilaments. They are found throughout, but one of the area where you find a large number of them is this area right here, and I'll grab my highlighter, where the axon connects. Because the axon has a lot of neurofilaments, so there's a large number of them, and our drawing kind of does a good job of showing these as well. As I mentioned, there are two types of processes. The dendrites are short and elaborately branched. There's really three key distinctions to a dendrite. They're short, they are elaborately branched, And while the picture doesn't demonstrate it, we know that this is where the cell receives information. So those dendrites are where the cell is going to receive the information. Conversely, our axon, as we've talked about, is single. It is unbranched, right? It is long, and it carries information away from the cell body. So basically, information comes in the dendrites and then travels out to the axons. And in this case, passes through the cell body. Now, the other thing about the axon, and this is probably a good place to mention this, so let's change colors to emphasize this. Our axon, where am I going to put this? I'll sneak it in down here. I'm going to put it down here just for lack of space. Axon has a specialized plasma membrane, kind of like what we saw in skeletal muscle. As such, remember, our plasma membrane of our skeletal muscle had a name. We called it a sarcolemma. Well, the same thing is going to be true here. Our axon's plasma membrane is therefore called the axolemma. Oops, why is that not? There we go. Can you guys see that on the bottom of the screen? Excellent. All right, perfect. Is the axolemma. And the axolemma, as we talked about, is specialized to produce and propagate action potentials. So, and I'm going to use my highlighter here to emphasize this, and we'll do it in red. All of this here, this part of the axon and everything beyond has that specialized plasma membrane, whereas the cell body and the dendrites don't. Well, it turns out it isn't just a barrier. This plasma membrane's totally normal, and this one's totally unique. Instead, there is a bit of a transition area. This transition area is this area right in here. And this area right in here is where the plasma membrane is changing. And this area where the plasma membrane is changing is what is known as the Axon Hillock. I'm going to sneak that in that way. So the Axon Hillock is that transition area. Where it is being changed from being normal plasma membrane to special plasma membrane. Now, if you think about it, those changes that are taking place, if we just change one small tiny component of it, it's not going to change the function. And then we change a little bit more and a little bit more and a little bit more until eventually we reach an area that has become so specialized that now it's capable of producing an action potential. Now, as we look at this Axon Hillock, we can't see where that point is, but we know somewhere in this Axon Hillock is an area where it has been changed enough, where the plasma membrane has been changed enough to now produce an action potential. And so basically, this is the first location on our neuron where we can produce an action potential, and that first location is what is known as the trigger zone. So inside the Axon Hillock is an area called the trigger zone, and this trigger zone is where we can produce our first action potential. All right. Questions on that? All right. Let's look at the other end. Oh, wait, hold on. At the bottom. Oh, actually, it's not supposed to be dot at all. It's supposed to be 2. I'm not sure how I got dot. Oh, that's what it is. This was supposed to say specialized 2. So the D ended up in the wrong spot and the O and the T ended up backwards. Thank you for catching that. So our axolemma has been specialized to produce and propagate action potentials. Yeah, you can call it trigger zone as opposed to what? The area where you first produce the action potential is the trigger zone, but it's within the axon hillock. So the axon hillock and the trigger zone are not the same thing. The axon hillock is the area of transition, right? That's the key. The axon hillock is the area of plasma membrane transition. And that one location in the axon hillock where it is finally changed enough to produce an action potential is the area that we call the trigger zone. All right, so does that differentiation make sense? Awesome, excellent. All righty, so any other questions about the proximal end of our neuron? It's gonna get easier when we get to the other end because those are all things that we have talked about. So any other questions on this before we switch ends of our neuron? All right, perfect. When we look at the other end, and I clear my pretty picture. Again, notice here we've got, again, these are the terms that were used. I don't think this is even from your textbook. I think this is from another textbook. Telodendria, which again, we are calling, and again, you're welcome to use the term telodendria. We are calling them the axon terminals. So those two terms are interchangeable. They use the term synaptic terminals. We're using the term synaptic end bulb, but again, they're interchangeable. And like I said, there are easily a half dozen more terms that are used for these as well. The key is that we have a single axon that branches into multiple branches when it nears the end of it. And at the very end, we have this specialized structure and as we learned in the muscular system, there are 2 main specializations to this synaptic end bulb. What are the 2 main specializations? What did we learn is special about those synaptic end bulbs? Well, we know they contain a chemical signal that they release to communicate to the target cell. And what was the generic name? There you go, that it is. It is that neurotransmitter, a neurotransmitter. There have been close to 100 different neurotransmitters that have been discovered and identified. We have talked about one of the most common ones, and of course that most common one is acetylcholine. So that's one example of one. But like I said, there are close to 100 different neurotransmitters that have been identified. So they can release acetylcholine. And of course, as we talked about, they're not going to release that neurotransmitter willy-nilly. We need a special way to tell them to release that neurotransmitter. And releasing neurotransmitter is a bit of a wonky thing. So what do we use? There you go. And not just any calcium channels, but remember our voltage-gated calcium channels. Excellent. So the second specialization of those synaptic end bulbs is our voltage-gated calcium channels. Those voltage-gated calcium channels let calcium into the synaptic end bulb, and when that calcium comes in, our synaptic end bulb is able to release its neurotransmitter, release that chemical signal to communicate with its target cell. So that is the anatomy of a typical neuron. Of course, as I'm sure you realize, the problem with that then is that typical means that all of them are not identically this way. But this is the typical anatomy. Any questions on this typical anatomy? Okay, now that we understand the typical anatomy, what we can do is we can classify our neurons. And not surprisingly, there are two ways we can classify our neurons. We can classify our neurons structurally, and we can classify our neurons functionally. So that's going to be our goal next. Now our goal is to, now that we know what a typical neuron looks like, we can talk about specific neurons, and we can talk about them based on their specific classifications. Clear all drawings. Structural classification. With our structural classification, and this is what we will start with, let us talk first about our structural classification. And as it says, structural classifications are basically identified by the number of processes that come off of their bodies. So let's draw 3 neurons here. And for the cell bodies, we'll keep them brown. There's a cell body, and there's a cell body, and there is a cell body. Excellent. 3 different centrally located cell bodies. The way we identify our structural classifications of our neurons, and since I've drawn 3 cells, it should not surprise you that there are 3 main structural classifications of neurons. And they're based on the number of processes that come off the body. And one of the important things to remember is that there are 2 types of processes. There are going to be, and again, I think for consistency's sake, let's go with color. There are going to be dendrites. There, and we'll use blue. Axon. All right, basically 2 types of processes. So how many they have coming off of them determines what type of structural classification of their neuron they are. Let's start first with, and we'll use big bold lines, this first one here. This first one here has 1 dendrite that comes off of it. Now, that dendrite is capable of branching and can receive information from many branches. But notice primarily, it is 1 dendrite that actually connects to the cell body. This neuron also is going to have one axon that comes off of it. And again, we know that axon is capable of branching at the end. And so it has 1 axon. This neuron, notice, has 2 cell bodies coming off of it. And since it has 2 processes coming off of it, and because it has 2 processes coming off of it, we call this a bipolar neuron. And notice with this bipolar neuron, information goes from the top of the screen down to the bottom of the screen. Because remember, dendrites receive information and our axons send it out. This second neuron is interesting in that what happens with it is that it has, rather than this one, which has a centrally located body, its body is off to the side. It has one axon that runs kind of parallel to the body. Of course, as we know, it can branch at the end. Oh, way too big. Way too small. There we go. Let's see. Always good. It has one axon off to the side. Now, obviously, it still needs to be able to receive information. So it needs to have dendrites. But in this case, the dendrites actually don't connect to the cell body. The dendrites actually connect to the axon. So what's unique about this one is with this one, the dendrites attach to the axon. So in this case, notice there is only one process that comes off of the cell body. And so this is what is known as a unipolar neuron. All right. Notice so far, none of these things look like the neuron we've already drawn. The neuron we already drew, and the one we've been familiar with since the get-go, has had many dendrites coming off of the cell body. They're short, they're elaborately branched. I've drawn 4, but really the key to this is that they are going to be 2 or more dendrites that are going to attach to the cell body, but still just one axon that, again, has the potential to branch at the end. This one, right, bipolar had 2, unipolar has 1. This has more than 2. So this particular one here is what we call a multipolar neuron. Notice this is the one that we have been drawing every time we said a typical neuron. And that's because from a structural standpoint, and actually let's do it this way. From a structural standpoint, and let's actually write it out. This is the most common type. So from a structural standpoint, multipolar is the most common type. Questions on that? But I know you have to have at least one. I have one. Maybe some of you that have looked ahead at Unit 12 might ask, all right, if you're all going to be quiet or you're all asleep, either is acceptable, I guess. I'll ask it. Well, what about the pseudo unipolar? All right, isn't that one of the terms that you may have seen in your textbook? The term pseudo unipolar. Anybody seen that term before? You can just give me a raise of hands on your participation or say yes in the chat window. There's one, there's two, there's three, couple more. Excellent. Yep, see, that was more what I was expecting. Absolutely. Well, where that term pseudo unipolar comes from is it's really just another name for these unipolars. What happens is that what we're looking at here is a mature adult neuron. What actually happens to these things in a developmental state, and I'll use purple to indicate this, what actually happens is it actually has, and here I'll cheat, I'll just draw a second one smaller next to it. There's our cell body. Let's go small. Perfect. What actually happens is it actually has two axons that grow out of it. One of those axons grows one way and one of the axons grows another. And eventually what happens is that these two, as it matures and grows, these two branches basically grow together and become a single structure. So technically it was two processes that started out that became one. And so that's what, of course, pseudo means. Pseudo means false or fake, as we've heard before. So technically your textbook, and your textbook uses the term, which is the only reason why I'm wasting 30 seconds of time talking about this, is because your book uses the term. But the term pseudo unipolar is basically the exact same as unipolar. If you like writing out pseudo unipolar, you're welcome to do that. But I would strongly encourage you to just simply use the term unipolar, because unipolar is the correct term. It's a developmental thing that honestly we don't care about. But technically that is where the name comes from. All of our unipolar neurons started with two small branches that fused together into one. So that's why they call it pseudo unipolar. All right. So those are our structural classifications. And while we're here, I'm going to go ahead and cheat. Let's take this and take this. And then what I'm going to do is hide all of that so that we can also talk about, and we'll use the color, what haven't I used yet? Green, to talk about the functional classifications of neurons. Now, the good news is that just like there are three structural classifications, there are three main functional classifications. And basically, we define by the direction it carries information. All right? In particular, in relation to the central nervous system. Those three classifications are, not surprisingly, sensory, which remember we called also afferent, and they carry information into the central nervous system. Our second is motor, or again, what we called efferent. And again, as we talked about, they carry information away from the central nervous system. So what does that leave us with? Well, what that leaves us with is basically neurons that carry information in the central nervous system, or we could think of these as being the neurons that are between the sensory and the motor. Those neurons that are between the sensory and motor, those neurons that carry the information in the central nervous system, we call our neurons. So interneurons are the neurons that carry information in the central nervous system. They're the neurons that carry the information between the sensory and the motor. And if you think about it, these are many of the neurons that are found in our brain and spinal cord, which is where the majority of our neurons are. Majority, 90% of our neurons, over 90% of our neurons are in the brain and spinal cord. So not surprisingly, and let's go ahead and do this. Interneurons are going to be the most common functional classification. All righty. Questions on that? Because we're not quite done. Let us cheat. I want to grab this, see if I can sneak that up a little bit. Perfect. All right, excellent. Now, those are our functional classifications. These are our structural classifications. And not surprisingly, there is a relationship between them as well. It turns out that most, let's put that in caps, most of our sensory neurons are unipolar. So if you randomly reach into the body and pull out a sensory neuron, it is most likely going to be a unipolar. Notice most is not all. There are a few exceptions, and that's where the bipolar come in. Bipolar are the most rare of the types, and they are only used in special sensory pathways. So you see them associated with the eyeball. You see them associated with the ears. You see them associated with smell, things along those lines, taste. Only are special sensory. The rest of the sensory are unipolar. And of course, that therefore means that all motor and all interneurons are multipolar neurons. And if you think about it, our multipolars are most common type, our interneurons are most common type. So if you were to take all the neurons out of a person's body and put them in a bucket, reach in and pull it out, the most likely thing you would pull out would be a multipolar interneuron. All right. Questions on that? Let me take a picture of this because that's pretty and it'd be fun to share that on the website. I've done this, and again, it looks truly amazing. But let me go ahead and clear this, and we'll go through it all again with the pretty pictures from your textbook. So as we talked about, our structural classification is based on the number of processes. Here, we see an example of a bipolar neuron. This bipolar neuron has a centrally located cell body, a single dendrite that feeds information into it, and a single axon which carries information out. And here, again, we can see we're starting to synthesize information. Notice our bipolar neurons are only involved with our special sensory pathways. Most sensory pathways use these unipolar neurons. Single axon, a cell body to the side, one attachment, and that axon travels down, dendrites at the top. And again, notice all of these carry information the same direction. Everything carries information. Oh, no, that doesn't help if it's white. All of these things carry information down the screen. And then, of course, our third Here's that typical neuron, centrally located cell body, 2 or more dendrites, 1 axon, again, carrying information down the screen, receiving it at the dendrites, traveling it down the axon. This is our multipolar, and it is the structural classification. Multipolar is the structural classification of all of our motor neurons and all of our interneurons. And then again, we can flip it around and do it the other way as well. The functional classifications are the directions we carry information. We have our motor, which is efferent, which is out of the central nervous system. Those are primarily multipolar. Our sensory, afferent, primarily unipolar. And then our interneurons, again, those found in the brain and spinal cord. They're the ones between the sensory and the motor. They're the ones that carry information in the central nervous system. And they're primarily multipolar as well. Of course, it can't all be just that simple. Not that that sounds particularly simple, but notice, for instance, here, we actually see 2 examples of what are multipolar neurons, right? Because that's part of the problem. Multipolar neurons can be both can be both motor and interneurons. So how do we tell them apart if they're both multipolar? The third component, location, and then also shape. What I have here are 2 multipolar neurons. Notice this one has 2 dendrites and a single axon. This one has 1, 2, 3, 4, well, let's go 1, 2, 3, 4, 5 dendrites and 1 axon. So both of these are multipolar. They have slightly different shapes. What's cool about this one, this cell is a Purkinje cell because Purkinje was a famous neuroanatomist who loved to use the microscope, was a beautiful artist. So what he would do is he would look under the microscope and he would do these beautiful, elaborate drawings of the things that he saw in the human body. And because he was an incredibly conceited ass, when he would publish these things, he would name them after himself. So there are over a half dozen different structures in the body that are called Purkinje. We are going to learn about Purkinje cells right now when we're talking about the nervous system. When we get to the heart, we'll learn about Purkinje fibers. He's all over the place. These, on the other hand, because they have this kind of pyramid shape to them, are what are known as pyramidal cells. These are both multipolar, and they're both interneurons, but they have different names because they're in different locations. This one is in our gray matter of our brain, in that cerebrum. We haven't talked brain anatomy yet, but we will. Here, I'll do a quick, horrible drawing of this up here at the top. Your brain, in simplest terms, looks like a big mitten. It has a big, huge part for the 4 fingers, and then a tiny little bit for the thumb. This structure that I've drawn has all sorts of wiggles and giggles and stuff like that in it. And this entire structure that looks like a mitten is what is called the cerebrum. That's the primary thing that we call the brain. And the gray matter, the large amount of cortex that's in this, is filled with these pyramidal cells. These are the cells that help you to make your decisions and help you to move your fingers and help you to talk and help you to do all sorts of other things. But when we look at the anatomy of the brain, the other thing you'll see is there is a tiny little nub, like a cotton tail, that sticks out the back. And that structure is known as the cerebellum. And oops, I hope so I spell it right, cerebellum. And that cerebellum is actually where you find these Purkinje cells. In fact, that's the first thing we'll try to look at histologically on Thursday when we come back. We're not going to get to it too much today, but when we start on Thursday, we're going to get serious about some of our histology, and we will talk about those cerebellar cells and those Purkinje cells that are found in there. So again, there are going to be lots of different types of all of these, and one of the main ways to tell them apart is going to be their locations. All right, so that is the anatomy of our neurons. We've looked at their anatomy, we've identified them structurally and functionally. We still have to talk about the physiology of how they function, but we're going to get back to that a little bit later. So any questions on the basic anatomy and the structural and functional organizations of our neurons. All right, nothing but blank silence. So from our neurons, and again, your book's got a nice table that talks about all these, looks at the different types and discusses these so you can get a chance to identify them. And again, notice even they use the terms interchangeably. What we need to talk about next then are those support cells. Like I said, there have been over 100 neuroglial cells that have been identified. You're going to be responsible for about 7. But this is another good natural stopping point. So let's go ahead and take our next break. We'll take another 10-minute break. So that means coming back at 2:27. And I will restart the recording at that time. All right, so any questions before I pause the recording and we take our break? Going once, going twice. All righty, I will see you guys in 10 minutes. So now that we have talked about our neurons, what we need to talk about are our support cells, our glial cells. And as I mentioned, there are 6, 7 that we are going to be responsible for. Actually, I think it's 6 now that I think about it. Ah, perfect, it is just 6. And the real key to remember for these is that with the neuroglial cells, there are different ones in the central nervous system versus the peripheral nervous system. So as you can see over here, there are 4 main ones, 4 main types we'll be responsible for in our central nervous system, and only 2 that are going to be found in the peripheral nervous system. So just a total of 6 neuroglial cells that you will need to know. So let's go ahead and go through them. Here are the lists, but let's go through each one's individually. Starting first with the astrocytes. As it says here, these are by far the most common glial cells in the central nervous system. You can see where they get their name. They have a big, huge, centrally located cell body, and then they have a large number of big processes that come out, so they look like big stars. As you can see, these help to form part of the scaffolding. They help to give some of the framework, right? So they're providing the support and the framework for our central nervous system, for the brain and spinal cord. So it's very important from a structural standpoint. However, they also metabolize our neurotransmitters. Remember, as we talked about, one of the things that happens is neurotransmitters diffuse out of the synaptic cleft. So we need to be able to metabolize those and break them down. They help us to maintain our ionic balance. In particular, they reabsorb excess potassium. Remember, we've talked about how potassium sneaks out while sodium goes in. And so we need to keep low levels of potassium outside. And all of those things that they do in and of themselves are vitally important. However, probably the most important thing that our astrocytes do is form what we call the blood-brain barrier. As you can see from this illustration, our astrocytes will take their processes and wrap them around the capillaries. Capillaries, of course, is where materials come into and out of. This is where materials are exchanged. And even though we haven't mentioned it, one of the things that hopefully you are aware of, let me go ahead and clear all the drawings for this, is that neurons do not regenerate. So if a neuron gets damaged, that's it. If it dies, I should say, it cannot be replaced. Right? And so we need to be really careful about what gets into and out of our brain's interstitial fluid. Basically, we need to filter and regulate what comes out of the capillaries. And that's what our astrocytes do. They help to perform a protective barrier to limit what types of things can get out of this capillary and have the potential to affect our neuron. Now, of course, it's not perfect. In particular, the astrocytes are just using their own plasma membrane. And as we know, plasma membrane is primarily made up of phospholipids. So things that are lipid soluble, like, oh, I don't know, alcohol. Does alcohol have any problem sneaking through this barrier, coming out and influencing your neurons? Yeah, absolutely. It has the ability to do that. So again, it isn't a perfect barrier, but it does help to limit what gets out and regulates what influences our neurons. All right, so again, super, super important from a functional standpoint that way. Let's talk about our second neuroglial cell, and those are the ependymal cells. Ependymal cells are a form, as it shows here, a form of an epithelial tissue. Epithelial tissues, of course, as we know, line surfaces. In this case, this lines the surfaces, and in particular, the cavities of our nervous tissue, of our brain. Our brain has space on the outside, and it actually has some spaces on the inside as well. These inside spaces are lined with these ependymal cells, and those spaces are filled with a special fluid. That special fluid is our cerebrospinal fluid. And so these ependymal cells play an important role in the production and the regulation of our cerebrospinal fluid. In fact, they're going to help to form the structures that make our cerebrospinal fluid. There's always that alphabet soup name that you know you can be guaranteed you're going to have to spell at one point or another. And in this case, that is the oligodendrocytes. Oligodendrocytes, if you notice, at first look similar to the astrocytes. They have a central located cell body. They have multiple processes that come off, and those processes wrap around things. However, the big difference between the oligodendrocytes, and the astrocytes is our oligodendrocytes are able to wrap around the axons, and only the axons of as many as a dozen. Oops. Different axons. And when they do that, what they do is they actually wrap their process, instead of just holding it in space, forming that scaffolding like the astrocyte does, our oligodendrocytes will wrap its plasma membrane hundreds of times around the axons. And when it does this, it basically produces this special insulation. And this special insulation is called the myelin sheath. Oops, I spelled sheath right. And notice that's what we see here in the illustration. This is that myelin sheath. It's wrapping itself hundreds of times around it, forming an insulation. Again, if you think about it, this makes some semblance of sense. If you look at the axons, they're like wires. And if you look at the wires in your wall, they have insulation around them to separate them. So obviously that insulation helps to isolate individual axons. like the wires in your wall. When you turn on the light switch in your room, you just want the bedroom light to come on. You don't want the garbage disposal to go on downstairs, or the garage door to open, or any of those types of things to occur. We want to isolate the individual axons. But when it wraps itself hundreds of times to form that special myelin sheath, this myelin sheath in particular serves a very important function. And that important function is that it speeds up the action potential. In fact, it can speed the action potential up to 6 times as fast. So it can dramatically increase the speed of our action potential so that that signal gets where it's going much, much faster. Again, these are the same words that are going to show up on the screen, but let's go ahead and do that anyway. So again, they form their myelin sheath. They can do it over a dozen different axons. And again, while it provides insulation, most importantly, it increases the speed of the action potential. Now, as it turns out, our axons in the peripheral nervous system are going to need myelin as well, but it will not be the oligodendrocytes that do that. Instead, it will be a different type of cell. And although we'll talk about it again later, I can bring it up now. If we start thinking about just how important is it to put this insulation to get this increase in speed, well, there are conditions in the body that where our immune system can break down and damage this myelin sheath that is produced by the oligodendrocytes. Right? Our body defense cells, instead of attacking other things, attack us, and they attack this myelin sheath, producing a whole lot of cracks and damage in this, and it disrupts the function of our axons. And anybody know what that condition is called? Where we get those many different breaks in our myelin sheath? Leukemia is not a bad guess, but the many breaks. Those are on VA are purposeful breaks that we want in there. Those are actually the spaces that are going to be functional. Now, multiple sclerosis. Multiple sclerosis is a neurological disorder where the oligodendrocytes are attacked and destroyed. The myelin sheath that is produced is attacked and destroyed by our own immune system. And that can affect your ability to move, to breathe, to be able to see. It can be an incredibly debilitating autoimmune disorder, multiple sclerosis. And these are the cells that multiple sclerosis targets. All right, let's clear that and take a look at our last glial cell, and those are our microglial cells. Microglial cells are actually more like phagocytes. These are more similar to white blood cells. Their job is to go to the site of an injury and basically destroy pathogens and get rid of dead cells. Now, one of the reasons we study the brain, we study the field of neuroscience, is to understand how things work. Because once we understand how things work, we can actually take advantage of that or try to manipulate it to our advantage. Here with these microglial cells is probably one of the most classic examples, right? Because these phagocytes, these microglial cells, are really good at their job. And as it turns out, they might actually be a little too good. Because when there is a traumatic brain injury, these microglial cells will go to that area and they will destroy all of the dead neurons. But they're not very discriminating. If there's an injured neuron, they'll destroy that too. right? Their attitude is kill them all, let God sort them out, right? Yeah, sure, you just have a bent dendrite, but you're probably going to die anyway, so I'm going to take you out, right? And so what happens is when we have that traumatic brain injury, a large number of neurons get damaged as a result of that, and it affects our cognitive function. UC Davis was one of the, and still is really, has a huge major neuroscience program there. And there were some researchers there. And again, you have to remember that I will start by when I tell this story, I will tell you two things. The first is that this occurred many, many years ago. So even if you were allowed outside, you can't go out and protest because you're not allowed to be anywhere near them. And these people are all long dead, and I'm sure they died all horrible deaths. Okay, so I will start with that right now. They all died horrible deaths, painful, slow, painful deaths, got what they deserved for doing these things. But the research that they did was vital for our understanding of how this process works. What they did is they studied rats. Rats are decent swimmers, but they're not the best swimmers. And so what they would do is they would take a big pool and they would put a rat in there. And I'll just put a rectangle there as my rat. And the rat would be able to swim around, but they're not very effective swimmers and they can't reach the bottom, so they tire pretty easily. So one of the things that they would do is they would take a plexiglass island, basically a box, and put it inside the pool. Because it was clear plexiglass, the rat didn't see it. So what would happen is the rat would be swimming around and swimming around and swimming around, and just randomly by chance, it would find that block and it would stand on the block because it wouldn't have to swim anymore and it would be happy. They'd take it out of the pool the next day, they'd bring it back, and when they brought it back the next day and they put the rat in the pool the next day, guess what the rat did? He swam right to the island so that he could stand on the island and he wouldn't have to swim anymore. And so what they would do is they would time how long it took that rat to reach the island. Then they induced a traumatic brain injury. The way they did this, because again, remember this is science, so they wanted to quantify these things. They had the rat, this will be our rat. And what they would take is they took this hollow tube and held it over the head of the rat. And then they would take a heavy weight and they would drop that heavy weight through the tube so that it was the same amount of force every time to produce a traumatic brain injury on that rat. Again, these people all died horrible deaths. It's okay. They're all burning in hell right now. But they would induce a traumatic brain injury into that rat. And of course, once they did that, they would then bring them back to the pool, put them in the pool, and not surprisingly, the rats did worse and it took them longer to find their way to the island. And that showed that there was a loss of cognitive function from the traumatic brain injury, right? You get a traumatic brain injury, you're not as smart, you're not as clever, your brain doesn't work as well. Right? Consistently shows us what we would expect to happen. Then what they did is the exact same thing. Only this time, immediately after they produced that traumatic brain injury, what they actually did is they gave a chemical, and that chemical suppressed the microglial cells. When they took these animals where they suppressed the microglial cells and they put them in the pool, what they found is that the rats were much better at being able to get to the island. There was much less damage to the brain. And it probably turns out because these microglial cells are so aggressive. Some of those neurons that would normally be destroyed by these microglial cells are not so damaged that they can't recover. And so if we're able to get those to recover, we can preserve some cognitive function. So if you think about the ways that we deal with traumatic brain injuries or traumatic spinal cord injuries, right? Even as little as 10 years ago, if someone in the NFL had a spinal cord injury, they were pretty much paralyzed for life. Right now, 2 years ago, there was a offensive lineman for the Buffalo Bills who was injured during the first preseason game, had a traumatic spinal cord injury, and he was walking before the end of the season. Right? Cold compression, the chemical suppression, all of these things. Now that we're getting a better understanding about traumatic and brain injury, and again, nobody likes the idea of torturing mice, but the information we gain from that tells us a tremendous amount about how our body works, how our body recovers. And by understanding those things, it allows us the opportunity to potentially improve. And so a lot of our improvements that we've seen on how we deal with these traumatic brain injuries came from comparative research that was being done. So again, I was not a part of that. Nobody at UC Davis is doing it now, so don't, don't protest me, don't protest UC Davis. And like I said, I'm sure all of those people died horrible deaths. So again, they got what they deserve, but we learned a tremendous amount about how the brain works because of it. And this is a great example. Often, once we learn how something works, again, our goal is to manipulate it. We saw that when we were talking about Botox, the botulinum toxin, and things along those lines. Once we understand how these things work, we can manipulate them to our benefit. All righty. Questions on that? All right, excellent. Let's clear that. With that, that is our fourth and final central nervous system glial cells. There are two in the peripheral nervous system, and we see them here. And not surprisingly, the two peripheral nervous system glial cells kind of do similar things to the central nervous system ones. That starts with our satellite cells. Our satellite cells are similar, not identical, but similar to the astrocytes, at least from the housekeeping side. They don't form a blood-brain barrier the same way, but they, as you can see in this illustration, and you can also see from that why they get the name satellite cells, because they surround the cell body like satellites revolving around it in orbit. They surround that centrally located cell body. And they do a lot of the similar type of housekeeping stuff. They play a role in helping to, let's write it out. They play a role in helping to metabolize neurotransmitters. They play a role in maintaining ion concentrations, especially potassium, but also calcium and sodium. Right, so they play a vital role in being able to do those types of things in a similar fashion to the astrocytes. Notice also here in our axon, we see that we have some myelination of our axon as well. And it is the Schwann cells that are responsible for providing that myelination. Now, there is one big, huge difference. Both Schwann cells and oligodendrocytes produce myelin. All right. However, remember, and here, I'll sneak it in here. Let's write this here. So when we compare Schwann cells to our oligodendrocytes, remember, our oligodendrocytes can form myelin on many neurons, axons. Schwann cells wrap their whole body around just one part of one axon. Remember to sneak that in? That did not work. All right, so let's shorten that. Oops. Like that. And then this will be for the oligodendrocytes. Excellent. All right, so they wrap their body. So as we look at this illustration, this is one Schwann cell. This is a second. This is a third. This is a fifth. This is a fourth. This is a sixth. This is a seventh. Notice here we see it in cross section. Here we see actually how it works. Here's our cell body, our Schwann cell with its nucleus and its plasma membrane. And notice it takes its plasma membrane and it wraps it around hundreds of times, forming this big, huge, thick layer that, again, we call that myelin sheath. Notice over here on the right, some Schwann cells will instead just isolate axons. All right, that isn't true for oligodendrocytes. So if you notice in this case, here's 2, 3, 4. Here, let me go back to my highlight. 1, 2, 3, 4, 5, 6, 7, 8, 9 different axons. Notice this Schwann cell separates and isolates each one of them. Again, remember, we want to be able to keep them electrically separated. We want to isolate them so that our signal doesn't cross. But notice the myelin is not wrapping, the Schwann cell is not wrapping hundreds of times. So this is not a myelin sheath. And these axons, even though isolated from each other, these axons are considered unmyelinated. We still want to insulate them. We still want to keep them separate, but these are not myelinated, and so because of that, they do not get that speed boost because these are not myelinated. They don't get that same increase in speed that the myelin provides. Great question. Yes, absolutely. That is, again, remember, these are also support cells, and you are correct. For instance, a great example of this is, as I think I've mentioned in this class before, had several knee surgeries. When I had my first, largest reconstruction of my knee, I had to have my ACL replaced and some repair to my MCL and some other damages, a 4.5-hour surgery, a tremendous amount of fun. And when I woke up from it, there was about a $50 size space to the left-hand side of my kneecap where the skin had no feeling at all. Because what had happened in that case is that the nerve that was carrying that information to my brain had been cut. Now I have feeling there. And the reason for that is with these peripheral axons, if their support cells, like the Schwann cells, are intact and the cell body is intact, there is the potential, again, it is a limited potential, but there is a potential for these axons to regrow. It's not a fast process. It took close to 2 years before I got feeling back on the side of my knee. But you're right, if the cell body is intact, if the support cells are intact, it is potentially possible for some limited regeneration. But again, that limited regeneration is only in the peripheral nervous system, where we have those Schwann cells, where we have those satellite cells. We don't have really any regeneration in our central nervous system. That's why there's such a strong push for stem cell research. Because if our own cells can't regrow, can we get other cells to grow and replace them? So yes, great question. Yes, absolutely. The satellite cells and Schwann cells, if still intact and the cell body still intact, can potentially allow that axon to regenerate. The neuron's not regenerating. If the cell body dies, that's it. But for some of the peripheral function, sometimes those nerves can grow back. And that's typically the advantage of surgery. With a surgery, they're taking precise cuts, limiting the damage that is done, and so that increases the likelihood, right? If you're riding a bike and you stumble and have a huge major injury and half your leg gets ripped off, then the chance of regeneration is more limited. Awesome question. Any others? All right, I do want to point out one more thing as well. Notice that while the myelin, actually I think I have another picture that shows this, but I'll go ahead and do it here for starters. Where's my pointy thing? There it is, excellent. Notice the Schwann cells cover most of the axon, but there are small breaks in the myelin. As we'll see, these small breaks are vital for its function, and these small breaks are what are known as the nodes of Ranvier. And nodes of Ranvier are incredibly fun to say, but you don't actually have to write out the of Ranvier part. You can just refer to them as the nodes, and that will be acceptable. All right, and like I said, I think I have one more picture that shows this. So as I said, right, those Schwann cells form the myelin sheath in our peripheral nervous system. Yep, here it is. So myelin sheath, again, hundreds of layers of the phospholipids and the proteins wrapped around the axons. And most axons are myelinated. And of course, the key word there being most, because most is not all. They are produced in the central nervous system by, again, the oligo dendrocytes, and in the peripheral nervous system by the Schwann cells. As I said, they provide that insulation, they provide that protection, but the big, huge key to them is that they speed up the action potential. And so that is huge. Like I said, it can be as much as 5 to 6 times as fast. And then, of course, notice here, we have 2 Schwann cells, and there's that node of Ranvier, or the neurofibril node. But again, we can just use the term node. Just calling that the node is easiest to spell and perfectly acceptable. All righty. Questions on that? Awesome. With that, we have the basic anatomy of these things in our pocket. Like I said, we do have some more histology we have to talk at, or look at, really. It's not, we're not adding too much to it, but we're going to talk some about that. But we will save that, excuse me, for the next class. We also have some basic anatomy we have to talk about. But for the most part, we have enough of the anatomy in our pocket now where we can start talking about how these things work. And so now what we need to start talking about is the physiology of our nervous tissue, of our neurons in particular. Because as we've mentioned, neurons are irritable, right? That's the key characteristic. They're irritable. They receive a stimulus and they respond. And they respond by producing an electrical signal. Before we said action potential, but in reality, the truth is it actually produces just a potential. Because what actually happens is that there are 2 different types of potentials that a neuron can produce. The first is our action potential, which we talked about. It is a big, and again, remember, as we talked about, positive signal. In fact, an action potential produces a signal that is on the order of about 100, and again, because it's a positive signal, a positive 100 millivolt change in a cell. Huge, massive signal. And it is a fairly set value. It is all or nothing, right? By all or nothing, I think of in terms of the light switch you have on your wall. Many of you are in your bedroom, right? Or in your living room or something like that, and you have that light switch on the wall, which is an all or nothing signal. If you put the tiniest bit of pressure on the light switch, nothing happens. A little bit more, a little bit more. Oh, hey, hold on, I have a question. No, Schwann cells do myelinate axons. They absolutely do. But remember, just singular, one small section of one axon. So let's go back real quickly. Hold on, let me clear this. Actually, I'm not going to bother clearing it, but let's go back. This is one Schwann cell. Actually, I like this picture even better. This picture here, every single one of these is a Schwann cell forming myelin sheath. They can also help unmyelinated axons. So they can help unmyelinated axons, but absolutely in the peripheral nervous system, the Schwann cells are the only cells that produce the myelin sheath in the peripheral nervous system. All right, so did that answer your question? Clarify that? Excellent. All right, so all or nothing. Like I said, if you put just the slightest bit of pressure on that light switch, nothing happens. A little more pressure, nothing happens. A little more pressure, nothing happens. But once you reach that critical point, and again, if you think about it again, we've talked about critical points before. That critical point is called a threshold. Once you reach that threshold on the light switch, boom, the light comes on. And think of it this way too. If you push that light switch twice as hard, does the light come on twice as bright? No, it's all or nothing. The light is either on or the light is off. And that's basically what an action potential is, right? You can't be half pregnant. You can't have your light switch in your bedroom half on and only have, the light only at half intensity or something like that. It is all or nothing. It has a threshold. It is an all or nothing big signal. And remember, as we also talked about, this occurs only along the axon. The axon is specialized. Again, the axon is the only part of the neuron that is specialized to produce an action potential. And remember, as we also said, and to propagate it, which is a fancy way of saying spread it. Excellent. But that still leaves us our cell body and our dendrites. And it turns out our cell bodies and our dendrites also can produce potentials, what we call a graded potential. I keep talking about the light switch because you're in your bedroom or you're in your living room or something like that. But maybe some of you are in your dining room, sitting at your dining room table. And when you look over at your wall switch, you see a big, huge knob. Your light isn't all or nothing. Your light is variable, where you can turn that knob a little bit and get a little bit of light, or you can turn it down and get a lot of light, right? We have one of those in our dining room, right? So if it's just me and my wife having a nice romantic dinner alone, we like to turn the light up nice and bright so we can see each other and interact with each other and talk and enjoy each other. Of course, if we're having a family dinner, I've got that light turned down as low as I possibly can so that the kids can't really get a look at the fact that I'm crying and weeping hysterically. I don't have to look at all the food and the crap that's on the floor or all the dishes they leave behind or anything else that goes about that. We have that variability to the switch. And that's what these graded potentials are. They're small, right? Graded potential is somewhere on the order. And again, it can vary. Again, it varies. But it is typically somewhere around plus or minus 2 millivolts. Oh, that's not a plus sign. That's a plus sign. So it can be positive 2 millivolts. It can be negative 2 millivolts. And that's one of the other huge keys to our graded potentials. They can be positive or negative. Remember, our action potential is only Positive. It's only positive. A graded potential can be positive or it can be negative. It can vary in size. On average, plus or minus 2, maybe it can be as high as plus 5, but it could also be as low as plus 0.5 millivolts. It is going to be variable. It's small, variable, and because it's variable, it can only travel short distances. It's not able to go very far, right? Remember, our action potential can go literally meters. You can travel over a meter, right? We talked about you having a neuron in the small of your back that goes all the way down to your big toe, right? So again, on you, that could be close to 3 feet in length. But you know who else has one of these in the small of their back that goes down to their big toe, right? Kevin Durant. And I believe last time I checked, he was like 75 feet tall, right? And so that means that neuron's axon in his cell body's got to be over like 40 feet long to get from the small of his back all the way down to his big toe. These things have huge differences and they can travel long distances. But our graded potentials are tiny. They're small little waves, and they only go short distances. But that's okay because they're only going to occur on our dendrites and our cell bodies. And our dendrites and cell bodies are kind of short. And the goal is going to be to get those graded potentials to the trigger zone. And if we can get a big enough signal to the trigger zone, we can produce an action potential. So these two potentials, the graded potentials and the action potentials, are going to work together to produce and send our signal. We start with graded potentials on our cell body and dendrites. And if we can get a big enough signal to our trigger zone, right, in that axon hillock, in that part where we're specializing our axon, then we can produce that action potential and we can send that signal long distances. And that's going to be our goal. Of course, to do that, we need to be able to understand how and why we produce these potentials. And the key thing to that to remember, and now I've got to move what I just wrote. So let's get this, where can I put that? Yeah, sneak it up here. I have a lot of spelling errors in that. Oh, we'll survive. To produce both types of potentials, both an action potential and a graded potential, there are two things we have to do. We have to have our starting point for a cell, and that starting point is the resting membrane potential. And we have to have ion channels so we can move those charged particles into and out of a cell, and that is going to allow us to affect a change. What we're going to do now is a little bit of a review of information that isn't necessarily entirely new. We may be looking at it slightly different from what we did before, and how we've looked at this before, but we're going to try to make some sense out of the starting point so we can understand how these things work. Right? When we talk about a cell, and let's go ahead and draw my cell here. As we've talked about, our cell is polarized. There is a difference in charge inside and outside of the cell. And anybody remember what we said the resting membrane potential of a cell was? There you go, perfect. -70 millivolts. Meaning that the inside of the cell is more negative than the outside of the cell, which is more positive. Right? Now, remember, when it's at rest, the cell is at equilibrium. And one of the most important things to remember And again, we talked about this way back at the beginning of the class, but the important thing to remember about it here is when the cell is at equilibrium, remember equilibrium means that there is no net change. There is plenty of stuff that comes into the cell and out of the cell. Lots of things move. The movement doesn't stop. What stops is that there is no net change. So for every positive thing that comes in, a positive thing leaves. For every negative thing that leaves, a negative thing comes in. There is no net change. And at this point, our cell is happy. Right? The cell is at equilibrium. The cell is not changing. The cell is happy. All right? We get this polarization because there's an unequal distribution of ions across the membrane, and that is inconvenient. So let me cheat and do this. Hold on a second. I don't know if that'll work. All right, I'm gonna cheat and try something. Where am I? Oh, it's not gonna be easy to change. Ah, never mind. Okay. All right. So there's an unequal distribution of ions. At least I can do is grab this, move it up, grab this, and move it up, grab this, and move it up. That's one word, net. There's no net change, right? It's like hopefully your bank account is, right? Well, hopefully your bank account doesn't have no net change, but at this point in time, no net change would be a good thing, right? You have a job where $100 come in, you have to pay $100 in bills, right? Money came in, money went out, but you have the exact same amount that you had before, right? So there's no net change, right? There's plenty of gross, so it's more almost like an economical term, right? There's gross change. $100 came in, $100 went out. But if you had $100 in the bank to begin with, you still have $100 in the bank after that. So your net wealth didn't change. There was no change in how much you had. And it's the same thing here. If one positive sodium comes in and one positive potassium goes out, the cell has different stuff in it, but it gained one positive thing and it lost one positive thing. So its membrane potential doesn't change, right? If 2 potassium leave and only 1 sodium comes in, now 2 positive things have left, only 1 positive thing has come in, and now I've lost a positive thing more than I did before, and I'm a little more negative, right? If I only have $100 coming in, but $200 have to leave, then now I've lost something. But if $100 come in and $100 leave, I'm the same, no net change. And that's what we mean by that. There's no net change. Everything it essentially is the same. Great question. Any others? All right, so to maintain this, the -70, we have an unequal distribution of ions across the membrane, and we kind of know that. We know there is more potassium inside the cell. We know there is more sodium outside the cell. We know there's more calcium outside the cell. We know there is more chloride outside the cell. So those are all things that we know. We talked about that way back when we were talking about cells. So again, no new information here, but just reminding us of what we've talked about before. And remember, there are plenty of leak channels. Sodium has a huge driving force and wants to come in. Potassium wants to leave. So remember, one of the things we talked about is we have that special sodium potassium ATPase. Let's change colors here so that we can emphasize this. There we go. with that sodium potassium ATPase, remember, it uses ATP. And it uses that ATP to basically kick 3 sodium out of the cell. And they're positive ions leaving the cell. And it also helps to bring 2 potassium in. Come in. So if you think about this, sodium comes into the cell and then is kicked back out. Potassium leaves the cell and then it is brought back in. So we're using energy to do work. And if you think about it, 3 positive things leave, but only 2 positive things come in. And that's one of the reasons why this cell stays negative. But again, at this point, it's happy. The cell is at equilibrium. There's no net change. And that's awesome because the cell is happy. But if the cell's happy, it's not doing work for us. If we're going to get the cell to do work, we need to change things. Oh, and there's my pretty picture that shows this. Potassium leaking out and the pump bringing it back in. Sodium sneaking in and then being kicked back out. So we have all of that. But we want it to do work. And that's the other key to this. As I mentioned, here at rest, the cell is happy. But the key is that at rest, when the cell is at its resting membrane potential, the ions are not happy. Oh, that's tiny. Let's make that bigger. Ions are like 2-year-olds. And what do 2-year-olds care about? Themselves. All they care about is themselves, right? If you put sodium in a beaker, it's going to spread out in that beaker until it reaches equilibrium. All ions care about is that their own equilibrium. So all ions want to be at their equilibrium point. They don't care what makes the cell happy. They don't care. Sodium doesn't care about potassium. Sodium doesn't care about calcium or chloride or the cell. All sodium cares about is itself. Sodium wants to be at equilibrium. And just as the cell has an equilibrium potential, our ions have an equilibrium potential as well. And so that's going to be the key. If we know what the ion's equilibrium potential is, we would know what makes the cell happy, what makes the ion happy. And that is what causes the driving force on our ions. All right, I've made a bit of a mess of this, so let's switch to the whiteboard and go over this again. So let's go to the whiteboard, clear all of that, and then start over again with our drawing. Here's our cell. And for right now, let's just focus on sodium. Our cell is at a resting membrane potential where it's happy, as we talked about, is at -70 millivolts. All right? But remember, what we care about, what we're going to talk about right now is sodium. Because sodium, being a 2-year-old, like all 2-year-olds, and it is a charged particle, wants to be at equilibrium, at its equilibrium. Right? If you remember way back at the beginning of this class, we talked about beakers, and in that beaker, we put a membrane in the middle. And on one side, we had 70% glucose. And on the other side, we had 20% glucose. And as we mentioned, if we then go and poke holes in our membrane, glucose will move. And in this case, which way would glucose move? To the left or to the right? All right. Glucose would move to the right. And it would do that till it reached equilibrium. And equilibrium at this point would be 45% on this side and 45% on that side. Any of that sound vaguely familiar to you guys where we've talked about that before? Excellent. So it sounds vaguely familiar. That's excellent. Excellent. Right? It kind of works in a similar fashion. At least it starts in a similar fashion. What we're seeing over here, and even though I put blue for that, I want to go back to red for the writing of this. Basically, what we called this before, if you remember, we talked about a concentration gradient. All right, that was the term. Or the other thing really we could call this is a chemical force. Both of those terms are acceptable. There is a chemical force. And sodium also has a chemical force. Right? As we talked about, there is, and I'm going to totally cheat. No, not cheating that. There is a large amount of sodium outside of the cell. And a very small amount of sodium on the inside of the cell. So just like glucose wants to go down its concentration gradient, sodium wants to go down its concentration gradient as well. It wants to enter the cell. And as we mentioned, we call this its chemical force because it is trying to reach equilibrium. But here's the big difference between sodium and glucose. Sodium is a charged particle. Right? It's a cation, right? Which means it's positively charged. Right? Oh, and for this, we should change colors to blue. So sodium is a charged particle. It's a cation. It's positively charged. And as we know, positive things, anybody who's played with magnets know, positive things like negative things. And do not like other positive things. All right, again, hopefully, like I said, hopefully everybody here has played with a magnet before, and this is not new information. When you're playing with a magnet, it's very hard to get the two positive sides or the two negative sides to go together, but the positive and negatives, they attract and go together. Well, conveniently enough, as we talked about, sodium is positively charged. The inside of the cell is negative. So there is going to be an attracting force into the cell for sodium. And this attracting force, because it is positively charged and the inside of the cell is negative, we call this the electrical force or the electrical attraction, right? Because that's what we're talking about here. So notice there is both a chemical force and an electrical force that is really trying to drive sodium into the cell. One of the things that we've learned about sodium, and our cells know about sodium and they use it a lot, is we get sodium really wants to get into the cell. And we actually use that to get sodium to do work for us. But like I said, what sodium really wants is to be at equilibrium. And remember, equilibrium means no net movement. Right? That is what sodium really wants. It wants to reach the point where the movement into the cell is equal to and opposite to the movement out of the cell. So where sodium is happy is when one of the forces in is equal and opposite to the other force out. Right? Where sodium is going to be happy is where there is no net change. Let's do this as a thought experiment. We're going to start to let sodiums into the cell. And rather than drawing Na over and over and over again, I'm just going to make little orange dots. That's a sodium, and then another sodium, and then another sodium, and then another sodium. All this sodium is coming in because it really wants to come in. Well, notice these are all positively charged ions. So notice as these are positively charged, what's going to happen is that these sodium coming in makes the cell more positive. And as more and more and more and more and more and more and more sodium come inside, the cell is going to get more and more and more and more and more positive till the cell reaches 0 millivolts. At this point, notice there would be no more electrical force for the sodium to want to come in. At this point, no electrical force. There's no charge to want it to come in. However, there's still a chemical force. So even more sodium enters. And as even more sodium enters, what happens is this electrical force reverses. Once the cell is positive on the inside, then our electrical force, and I'll just abbreviate it, is going to be out. And let's make that a little smaller so it fits. And so now our electrical force is an outward force. We have our chemical force coming in, our electrical force going out. And notice also, text. As more sodium enters, the electrical force gets bigger until we reach a magical point. And that magical point is where the inward force, inward chemical force, and the outward electrical force are equal and opposite. It's a little tiny, so let's make that a little bigger. And that magical point right here, where those are equal and opposite to each other, is going to be equilibrium for sodium, right? We call that point the equilibrium potential of sodium. And that point is positive 66 millivolts. So if we could make the inside of the cell positive 66 millivolts, sodium would be happy. Again, the cell would not be happy at this point. Calcium wouldn't be happy, chloride wouldn't be happy, potassium wouldn't be happy. The only person that would be happy is sodium, but sodium is all that sodium cares about. the whole point of all of this, I know even though I can't see any of your faces, I know all your eyes have glazed over at this point. All right. But here is the take-home message. If you learn nothing else about the rest of this, here is the 100% important point to remember. The take-home message of all of this is that ions move to reach their equilibrium potential. All ions want to do is reach their equilibrium potential. And so if I open a channel, that's what's going to happen. So if I were to open a sodium channel on this cell, sodium is going to move into the cell to try to get the cell to positive 66 millivolts so that it can be happy. And it will keep coming in until it reaches that point or until you close that door and don't let it come in anymore. So really the whole point of all of this is to get to this take-home message. If we know the equilibrium potential of an ion, we know how it's going to move. And now we know sodium's. We know sodium's equilibrium potential is positive 66 millivolts. If you've noticed, there haven't been a lot of numbers I've made you memorize in this class. These are definitely some of them. The resting membrane potential of negative 70 millivolts, our equilibrium potential of sodium, right? If you remember back to the muscle, there was a threshold. Do you guys remember what the threshold value was? Anyone? Again, I appreciate normally the eyes have glazed over at this point. Come on, someone's got to remember. What was the threshold? There it was, negative 60 millivolts. These are numbers that are vitally important. And our equilibrium potentials of our ions are important as well. Sodium's is positive 66 millivolts. And even if none of the rest of this makes any sense to you, all you have to know is that if I open a channel, sodium's going to enter the cell to try to make the cell more positive. That's the take-home message from this. Great question. So the other ion that we need to know the equilibrium potential for is potassium. And I will save you the grunt work of getting to that point by just telling you what that is. So let's go ahead and go back to the lecture. All right. Again, to get to our equilibrium potentials, we need to know the chemical gradient or force, the electrical gradient or force. And as we mentioned, where's my annotation tool? For sodium, it is positive 66 millivolts. So if we think of our cell, we know the inside of the cell is negative 70 millivolts. So if we open a sodium channel, we know sodium, which is a positive ion, will enter to make the cell more positive because it is trying to get to negative 70 millivolts. So sodium will enter the cell to try to make the cell more positive. It turns out potassium, the equilibrium potential of potassium is negative 90 millivolts. And you may not have known that, but what you do know is that the cell at rest is at negative 70. Potassium wants to be at negative 90, so it wants the cell to be more negative. So if we open a potassium channel, which way does that positive potassium flow? Does positive potassium flow out of the cell or does positive potassium flow in? Potassium is going to leave, absolutely. If we open a sodium channel, right, our potassium, pardon me, if we open a potassium channel, potassium leaves the cell to make the cell more negative because it wants to be at negative 90 millivolts. And again, sodium enters, potassium leaves. That we already knew that. We already knew that was the way they wanted to go, but now we know why. It's because they want to be at equilibrium, and these are the equilibrium potentials we need to know. Other ions, other than sodium, potassium, we don't have to worry about. So sodium, potassium are the only equilibrium potentials we are going to worry about because the other ones are more complicated. So let's say it that way. But these we need to know. All right. Questions on that? Well, because remember, all they care about is themselves. So the question was, how does this work if they're doing the opposite? And in fact, if you think about it, this is why we need the sodium potassium ATPase. We need that sodium potassium pump. Because think about it. Let's change colors, black. If one sodium came in, right, if one sodium entered into the cell and one potassium left the cell would still be happy. One positive thing came in, one positive thing left, the cell would still be happy. But we lose our concentration gradients. Sodium would come in, potassium would leave, and we wouldn't be able to do work anymore. That's why we need that pump. All right, fine. The cell may be happy because one sodium came in, but as soon as that sodium came in, we need to then kick it back out again. Yes, that potassium left, but as soon as that potassium left, we need to bring it back in again. And again, to do this work, we need that ATP. So this is how the cell maintains equilibrium. Yep, you're right. And again, it is going to kick 3 sodium out. It is going to bring 2 potassium in. And again, 2 positive things come in, 3 positive things leave. And that's one of the ways we keep the inside of the cell negative. Yeah, that unequal movement of the ions is how we keep the inside of the cell more negative. Does that make sense? Awesome. All righty, let's go ahead and clear that picture. Oh, great question. No, it doesn't eventually get too negative because what you have to remember is what we're looking at here is just 3 things. What you have to remember is that there are hundreds of different types of leak channels and other ions that are coming in. Magnesium is moving back and forth. Calcium is moving back and forth, right? There's all sorts of other ions, chloride, all these other ions are moving as well. And so all of them together, right? All of them together is what equals equilibrium. It'd be kind of like a city. If you think about it in terms of a city, there are millions of people in the city. Some of them are going to leave the city to go to work. Some of them are going to come in. All right, some of them are going to die. Some of them are going to give birth. The actual individual people inside that city are constantly changing. But if the number of people that leave and the number of people that enter always stay the same, there may always be 1 million people in that neighborhood, even if one person leaves, but then another person gives birth. Boom, we have the same number. We still have 1 million. A new person moves into the area and another person dies. Right? Or 2 people leave and another person, 2 more people come in. You're getting constant movement of all these different ions, but together there is no change. And that's the key. Lots of movement, but no change. That's really what equilibrium is. Lots of movement, but no change. All righty. Now, as I mentioned, to be able to move these, we need our ion channels. That is how these things are going to do work. This is not new information, but it's information that I want to make sure to emphasize and go over again. One of those, as we talked about, are our leak channels. So when we have a cell, on that cell, there may be some channels that are constantly open. allowing whatever ion it is, one of those may be a sodium channel, allowing sodium to constantly come in, or a potassium channel, or a calcium channel, or whatever it is. These leak channels are always open. And that's why, as we've talked about, we need to use pumps to use ATP to get rid of things or gain things back that sneak in or out. So we use those pumps to offset the free movement of ions. And again, equilibrium is when those pumps and the free movement of ions are equal to each other. It's what makes the cell happy. It's what keeps the cell at equilibrium. But like I said, it doesn't allow us to do work, because to do work, we have to be able to change. And that's the key. Leak channels do not change. And if we can't change them, we don't do work. All right. Again, normally I use the example of campus, right? During the day, the gates to the parking lots are completely open, so people can come and go freely, all right? But the door to our classroom is not always open. If we want to do work, we need to open that door up, and that gets us inside, and then we can do our work. So to be able to do work, what we have to be able to do is we have to be able to open and close channels. And channels that can be open and closed are what we call gated channels. All right? These are the ones that can change their state. Right? They can be open or closed. Or maybe even more states. And it's these that we can change. They can be changed. And that allows us to do work. We may not have thought of it in these terms, but you're totally aware of this, because this is exactly what we did at the neuromuscular junction. In the synaptic M bulb, we opened voltage-gated calcium channels. Calcium came in, and that calcium coming in allowed the synaptic M bulb to do work. It was able to release acetylcholine. That acetylcholine, a chemical, then bound to a chemically gated channel. Open that channel, allowed sodium into the motor end plate, and that sodium entering into the motor end plate made the cell more positive, and it was able to fire an action potential, that muscle action potential. So notice, in our last section of the class, we saw two examples of where if we opened a gated channel, we could do some type of work. And that's the key to our gated channels. They can be opened, and by being able to selectively open them, we can do work. And we talked about two types of gated channels, voltage gated and chemically gated, but it turns out there's actually a third, mechanically gated, which, believe it or not, we've already talked about as well. We didn't use this fancy name for it, but we absolutely talked about it. Let's look at the pretty pictures for these, because I love these pictures. These are very simplistic pictures. I'll show you the pretty one from your textbook in just a minute. Yep, proteins are channels, absolutely, protein channels. A protein is, again, well, if you remember back when we talked about our plasma membrane is that phospholipid bilayer. And as we talked about, one of the things that will be in it is proteins. And some of those proteins are proteins that will form a channel. And that channel will allow substances to flow into or out of it. So these, and that's what these purple blobs represent. These purple blobs represent proteins, and this blue line represents our phospholipid bilayer. So this is essentially what our chemically gated cation channel was. Notice it starts in a closed state, and our poor little positive ion can't get through. But when a chemical binds to it, and look, conveniently enough, that chemical is acetylcholine. When acetylcholine binds to it, it opens, and once it opens, ions can freely flow through. and those are our chemically gated channels. All right. Remember, we also talked about voltage-gated channels, like our calcium channel. Remember, at threshold, pardon me, at resting, negative 70 millivolts, it is closed. But when we reach that magical point, and again, that magical point is threshold, and that threshold is negative 60 millivolts, then that gate opens, and our ions could flow. Now notice this one is closed. This one is open. But there is something interesting about our voltage-gated channels. Some, keyword there being some, voltage-gated channels have a second gate. Notice here and I'll highlight this, notice here on our illustration, they just drew it as another door-like structure. In reality, that's not what it looks like. So in reality, let's go ahead and put our plasma membrane back there again. Draw our two proteins. Here is a protein, here is another protein. And again, they have these gates that keep it closed. But there is actually a second gate, what is called an inactivation gate. And this inactivation gate is made of two proteins. One is a long linear protein, and the other is a big globular protein. And they give this inactivation gate a very technical name. It is referred to as the ball and chain. What's interesting about this ball and chain is the ball and chain and I have to cheat and do this, so let's open my activation gate so the channel is open. What can actually happen with this is when it's in this open state, it's not very stable. And what will actually happen is the ball of the ball and chain will actually come in and wedge itself into the channel. Notice when it wedges itself into the channel, ions can't flow through it anymore. It's still open, but ions can't flow. And when that occurs, we say it is inactive. Here we see it with the simple illustration from this simple diagram, that inactivation gate is closed. Notice the gate is open, but ions can't flow, it's inactive. But what actually happens is this ball wedges itself in there. And what's interesting about these ball and chain inactivation gates is once a channel is inactive, it cannot open again. Now, as it turns out, not never, but it cannot open again until it closes first. What that means is that, once it is inactive, that channel has to close first. And once it closes, then it can open. And once it opens, it's going to inactivate. And then once it's inactive, it has to close again before it can open. The reason for that is what actually happens is that these opening gates, what happens is when they close, they shove the ball out. And so what happens is the ball gets popped out by that inactivation gate when it comes back down. So when that inactivation comes down, it pops the ball back out. Your book has a beautiful illustration that does a really great job of describing this, and I will show it to you in a minute. But the key to note is that some of these voltage-gated channels, some, not all, but some voltage-gated channels have this second channel, this inactivation gate. So if you think about it, no, great question. No, it is not. Hold on one second. It is, and I'll answer that more thoroughly. It has 3 states: open, closed, and inactive. No, to answer your question, it turns out that both the ball and the activation gates are both positively charged. All right, because if you think about it, they open when the cell gets more positive. So when the cell gets more positive on the inside, these things don't want to be on the inside anymore, so they try to get away. I'm going to go ahead and erase this, and I can show you. Let's jump ahead. Whoops, not what I wanted. And look at the great picture from your textbook, because your textbook does a really great job of showing this. So here it is. Notice here is the actual protein, and here are the gates. These are the activation gates, and this is the inactivation gate, that ball and chain. When the cell gets positive on the inside, again, the key is it reaches threshold. That reaching of the threshold causes the gates to swing open. And when it swings open, now the channel is open and sodium can enter. But notice this ball of the ball and chain just randomly wedges itself in there. When it wedges itself in there, now the channel is inactive and the sodium can no longer enter. And the only way to get rid of that ball is to close the gates. Notice when the gates come down, they kick the ball out. And once the ball is kicked out, once the channel is closed, then it can be opened again. So this can only go from closed to open, open to inactive, and then once it's inactive, it has to close again before the channel can be opened a second time. All right. So again, I think this picture from your textbook does a much nicer job of showing that, representing that. Questions on that? Great question. That is an awesome question. Well, I'll tell you what, I will give you the honest answer. It's a little too early for this, but we'll go ahead and do this now. If you remember correctly, when we talked about our muscle cells, one of the things that we said is that when the muscle cell fired an action potential, there was a brief period of time where it could not fire an action potential again. We called that the refractory period. And as we talked about, that refractory period plays an important role in determining how quickly we could stimulate it. So notice skeletal muscle had a very short refractory period, and that allowed us to add a bunch of waves together and tetanize the muscle. Whereas remember, our cardiac muscle has a very long refractory period. And that long refractory period stops it from being tetanized, stops us from putting it together. What this inactivation gate does is it stops sodium from coming in again. It stops us from producing another action potential. So this inactivation gate is actually what determines our refractory period. Well, again, it is a physical barrier. That ball is literally a physical barrier that stops sodium from being able to enter. So yes, so technically the other gate has swung open, but when you wedge the ball in there, and again, this is usually when I demonstrate this in the classroom. So think of it this way. If we were standing in the classroom, what I would do is there's that door to the lab tech area. I would open that door, right? So first the door is closed, then I open the door and it is open. Once it is open, I then take that red garbage can that we had right there and I stick it in the doorway. Once I stick it in the doorway, the door is still open, but I can't get into the lab tech area because that garbage can is in the way. And that's what it means to be inactive. To have that garbage can stuck in the doorway stops you from getting through that doorway. And as it turns out, what has to happen is that door closes again. When the door closes again, it pops out the garbage can. And once it pops out the garbage can, then we can open the door and it can be open again. All right, so this is what happens. And the reason, again, action potentials are a current. Current is caused by a charged particle moving. Sodium is a charged particle, so sodium entering the cell is actually what produces the action potential. So if we stop sodium from entering the cell, we stop the action potential. Great questions. Any more on this one? Awesome, that's what I love to hear. All right, well then let's sneak back to the one thing that I skipped. And that is our third type of channel, a mechanically gated channel. As I mentioned, we actually already talked about this once before. If you remember in the skin, we talked about those Pacinian, or what we called the lamellated corpuscles that were made up of many layers of many layers of plasma membrane. Remember, we said they kind of had a onion-like appearance to them. Let's go ahead and draw a couple more just for the heck of it. All right, they had kind of these onion-like appearances to them. And there, remember, are the sensory receptors that perceive pressure. Right? As we talked about, when they are squeezed, we feel that sense of pressure from squeezing a ball, or we're feeling it on our back or our butts from the chairs right now. And as we talked about, it was that stretch of the plasma membrane that stimulated and gave us that sense of pressure. And that's exactly what happens with a mechanoreceptor. Notice here, at rest, the plasma membrane is in its normal position, and that gate is closed. But when we stretch the cell, or we push on the cell's plasma membrane, that mechanical force, and that's really the key to this, this is a mechanical force. That mechanical force is going to move the plasma membrane, and when it moves the plasma membrane, it opens up the ion channel, and ions can flow. And as soon as that force goes away, it closes again. So notice there, with the exception really of this inactivation gate, there's really no new information on the screen right now. We know about chemically gated channels, we know about voltage gated channels, we even know how they work because we used them in the last section. And while we didn't use the term mechanically gated channel, we do get the idea of when something stretches or when force is put on it, it can fire an action potential, and that's because it has mechanically gated channels that open up these channels when the cell is, when a mechanical force is applied to it, when it's pushed on or when it is stretched. All right. Excellent. All right, push the wrong button. Sorry, I got too much stuff on the screen right now. Clear the drawing, that's what I want to do. Excellent, all right. So, take-home messages. As we talked about, ions are 2-year-olds that only care about themselves. They are going, when we open a channel, a gated channel, when it opens, an ion is going to move to reach equilibrium. It wants to be happy. All that ion cares about is itself. And in particular, the two we care about are sodium and potassium. If you open a sodium channel, sodium is going to move to bring the cell to positive 66 millivolts. That is its equilibrium potential. Potassium, if you open a potassium channel, potassium is going to move to bring the cell to negative 90 millivolts. That's where it'll be happy, and that's all it cares about. And so if we use those gated channels, we can allow charged particles to move. And really, if you think about it, when a charged particle moves, that is a current, right? I know this is in a physics class. We're not going to talk too much about electricity, but as we talked about flipping that light switch to turn on the light, you flip that light switch and an electron moves along a wire. A charged particle moves from one place to another, and that is the current that allows your light to be turned on. And that's how our cell does work too. A positive charged ion entering or exiting the cell, a negative charged ion entering or exiting the cell is a charged particle that is moving either into the cell or out. And when a charged particle moves, that is a current. And those currents that we produce are going to be our potentials, our graded potentials and our action potentials. And that is where we're going to pick up on Thursday. On Thursday, we will, now that we have these basic building blocks, we'll try to make some sense of it. I know this was a little dense, but when we see it work on Thursday, hopefully it'll make more sense. We will talk about how we produce a graded potential, how we produce an action potential, and hopefully we can make some sense of that. All right? Questions on that? All right. We have covered a lot today, and I know you're probably exhausted and brain dead. There is one last thing, one last challenging thing that we do need to briefly talk a little bit about. And again, I'm not going to go too much in depth. I'm just going to introduce you to this idea today so that we can cover it in more detail on Thursday when we start talking about these things. So what is it that I want? I want... that's what I want. Perfect. Hopefully, because you've had 5 whole days since the exam, you have been looking at your brain anatomy list. Hopefully, most of that stuff has been making at least some semblance of sense to you, or you've been at least able to find to it. But the one thing that may be a tiny bit confusing if you've been paying attention is that on that list are terms like cephalons. These terms, cephalons, are basically just developmental divisions. If you think about it, when we learned the bones, we divided the body's skeleton into more easily digestible pieces. Axial skeleton, appendicular skeleton. And even within the axial skeleton, we did skull, we did vertebral column, we did bony thorax. When we did muscles, we did the muscles that move the head, the muscles that move the trunk, the muscles that move the arm, and so on and so forth. And so for our brain anatomy, we are going to divide it into pieces as well. And like before, we're not going to do it randomly or meaninglessly. But the way we are going to do it, interesting enough, is from a developmental standpoint. When we first start developing as a bundle of cells, and I need my annotation things for this, our nervous tissue undergoes this really interesting development. It starts first just as a flat sheet of cells called the neural plate. That neural plate then actually gets a little bit of a bend in it called the neural fold. That neural fold allows it to start to curl up to form a structure we call the neural crest. And that neural crest probably eventually forms a hollow circular tube of cells that we call the neural tube. And that's what we are showing right here. Right here, this bumpy structure is our neural tube at 3 weeks of development. Very, very early in the developmental process. This fluid-filled tube of cells continues to expand to form, as we can see here, structures that are called vesicles. And notice there are 5 main vesicles. This occurs as 6 weeks of development. Very, very early in the body's development, we start to get this specialization to the nervous tissue. And it may look like meaningless bumps, but as it turns out, even at 6 weeks of development, the cells of this region are already specialized and are going to become one and one thing only. The cells of this region are specialized, they're going to become one and one thing only, and so on and so forth. So these 5 vesicles, or more specifically, these 5 cephalons, oops, did I spell it rightly? Are important anatomical distinctions. And like I said, you can see there are 5 of them: a telencephalon, a diencephalon, a mesencephalon, the metencephalon, and the diencephalon. These 5 specializations, these 5 really, they're not even specializations at this point. These 5 blobs, these 5 enlargements become distinct parts of your brain. The telencephalon becomes your mature cerebrum. Notice the diencephalon and mesencephalon, these actually keep these names. The diencephalon is called the diencephalon in adults. The mesencephalon is called mesencephalon in adults, although I will admit sometimes the mesencephalon is sometimes referred to as the midbrain. So that would be an acceptable term for that as well. The metencephalon becomes the enlargements that are the cerebellum and the pons. And your diencephalon becomes everyone's favorite, the medulla oblongata. These 5 blobs that form at 6 weeks of development ultimately lead to here. We see, and notice they've color-coded it here. And these are pictures straight from your textbook. So your textbook has this as well. So here in the peach, we have the telencephalon. The pink is the diencephalon. The green is the mesencephalon. The 2 enlargements, the pons and the cerebellum, are the metencephalon. And then the orange is the medulla oblongata. So here is what that mature brain looks like. And here we see, and of course, these are highlighted because they're internal. If we cut the brain in half, then here, this part here is the diencephalon. This part here is the mes. This part here is the met. And this part here is the my. So to try to make more sense, to divide the brain into more easily digestible pieces, we will use the cephalons as our dividing point because they're meaningful in their divisions. So we're going to talk about all the telencephalic structures, all the diencephalic structures, and so on and so forth when we talk about the brain. All right. The last thing that I want to remind you about is, again, obviously, we are not going to be in the classroom. So there are not the models in the classroom to look at in the classroom, but you have a lot of great locations where you're able to see these things. Hold on one second. Oops, wrong button. That's the one I want. Remember, on Cosumnes River's site, there is that virtual anatomy lab. When you look here, and as you can see, I was already looking at it, the histology is really good. The brain models are really good. So here's the chart of the brain that we've looked at, that good half head we looked at so many times and we'll continue to look at. And they have a ton of stuff labeled for the brain models and the cranial nerves and all sorts of amazing things that they're going to be on here. This is a great, great resource that I strongly, strongly encourage you to take advantage of. Let's go all the way back here. This is just easier to do that way. There is some good histology on here as well. The other resource that you should be taking advantage of, yeah, obviously we're not going to be able to do a sheep brain dissection. However, if you happen to have sheep and you want to do a craniotomy, I can't stop you, but I cannot provide you with sheep brains or to be able to do that for that. However, as you saw, that visual anatomy does have a sheep brain that you can look at. So there is a dissection, so you can observe it, but there isn't any way to do that. The other thing that I wanted to show you, let me find it again. Perfect. Remember, on your modified mastering A&P, you've got that practice anatomy lab. And if you go to the practice anatomy lab, under the anatomical models, there is nervous system. We have central nervous system, peripheral nervous system. There are some great images. Now, again, some of these are pretty basic. But again, here is basically what our half head looked like. And it has some great anatomy where you can show the labels and remove the labels. You can go through all of this and again, show the labels. Oops, not close. Show the labels. We can see all of these things as we go through them, some different models. So there are lots of ways to study this material, even though you don't have an external brain you can play with at home. So even though you don't have that external brain you can play with at home, There are a lot of resources online available to you to start working on that anatomy. We will go over some of it together, especially some of the more challenging stuff and the things that I want to make sure that you understand. But you are responsible for knowing all of it. It is going to be a PowerPoint presentation. Less anatomy on this than the last exam. This exam normally is somewhere around, I don't know, 60 to 70 questions, depending on how far we get and how much of the sensory stuff we get into. So if we get into the sensory, excuse me, we'll be closer to 70. If we don't get into as much sensory, we'll be closer to 60. And like I said, there are some great handouts on this section for you as well, including a cranial nerve handout. As I mentioned, that cranial nerve handout could be as much as 25% of your lab exam. And we definitely will be doing it together. It looks like I've got right now scheduled for April 30th. April 30th is when I anticipate we'll be able to go together through our cranial nerves. And like I said, those cranial nerves could easily be 25% of your lab exam. So I encourage you to start looking at that information. Your book does a spectacular job of showing and talking about the cranial nerves. So make sure you take a look at that. But I also guarantee you, we will go through that handout together and make sure that it makes sense. So we will work on it together. But make sure you do that. So that is your game plan. Again, I don't have time to go over the anatomy today. I know you're ready for me to stop talking, and I will. But we will start working on the anatomy, the lab portion, on Thursday, because our lecture won't be quite as extensive. But we've laid the groundwork today, and so then we can start building on Thursday. All right? Questions on any of that? I'm sure, like me, you guys are all very tired, but I still have more grading to do. So hopefully, I got to go do work. You got to go do work too. Make sure you study hard and prepare yourself for Thursday. And then, like I said, I hope to have all the grading done by Thursday. I'm still not done with my 430. I'm trying to get theirs done as well. So I will be doing the best that I can to get everything back to you guys in a timely fashion. And again, as I said, be warned whenever, and again, we'll talk about it more on Thursday because you won't get it before Thursday. But either Thursday after class or depending on how far behind I am, sometime after that, your exams will be posted. So again, you won't get it before Thursday. I may post the grades, but you won't see the exams before then. But it would take a lot of luck for me to get that far before then. So we'll see how it goes. All right. I've given lots of people plenty of time. Any more questions, any more typing, anything like that, or are we just all ready to go? All right, I guess we're all ready to go. I will go ahead and stop the recording. Have a great day. Have a great week. Be safe. Take care of yourselves. And I will see you on Thursday.